because we have power interruption. So at least I can show my face a little bit. So good morning, everyone. Welcome to the 2021 IUPAC Global Women's Breakfast with the theme, Empowering Diversity in Science. I am Dr. Gladys Completo from the Institute of Chemistry, University of the Philippines, Los Baños. And for this event, with me are my co-host, Dr. Darlene Dalisay from the University of San Agustin and Dr. Cheryl Ariola from the Institute of Chemistry, UP Baños. So this event is hosted by the Philippine American Academy of Science and Engineering, or PAASE. PAASE is a non-profit organization composed of scientists and engineers of Philippine descent who have distinguished themselves in scholarly and research-related activities. PAASE members are at the forefront of scientific research and technology development in the United States, the Philippines, and other countries. Since 1980, the organization has grown to over more than 250 members. Our mission is to contribute to national progress through the improvement of the STEM ecosystem in the Philippines. So today we celebrate the 2021 IUPAC Global Women's Breakfast, focusing on the achievements and contributions of women across the world. Women are as citizens, mothers, daughters, leaders, and scientists. Women who, just like men, are trying to make a difference in this world. I would like to officially start this event by sharing a video from the IUPAC organizers. Let me just share screen. Can you see the screen? Doralian?
Yes, Gladys, we can see the screen. Yes.
Can you see it?
Yes, ma'am.
Yeah, there.
Laura McConnell from Bayer Crop Science. And with me is Dr. Mary Garson from the University of Queensland in Brisbane, Australia. We are thrilled to welcome you to the 2021 IUPAC Global Women's Breakfast.
It's our very great honour to kick off the 2021 Global Women's Breakfast, Empowering Diversity in Science. We know many of you were there with us back in 2011 when we hosted the very first Global Women's Breakfast. Thank you. And also welcome back to those who participated in 2019 and 2020.
This year, we want to extend a special welcome and thank you to the organizers, the men and women who have stepped forward in their organizations, and also to those who are attending the Global Breakfast for the very first time.
We love seeing all types of organizations participating. That includes universities, scientific societies, industry, the government sector, and most importantly, high school classes of aspiring scientists. And there's always so much excitement. The very first breakfast starts in New Zealand, of course, and on the global map on our website, from that moment on, we see a sea of red hearts lighting up as breakfasts start around the globe.
It's really your leadership and enthusiasm that has inspired IUPAC to make this an annual event. If we look back at the previous breakfast, chemists from all over the world, from more than 50 countries, joined in. In both 2011 and 2019, we were really celebrating, and rightly so, achievements of women chemists. In 2020, we started developing a true global network. And now this year, we're ready to put it into action.
So today we're asking groups to connect with each other, to strengthen the bonds that have already been created, and to extend those networks to include new groups and organisations. And this is so important in the current COVID world, where many people have been feeling isolated.
That's right, Mary. And looking at the many groups who have developed programs for their events, some are highlighting, women leaders in their organizations. Some are having conversations on the challenges that women face in their careers, and others are supporting young scientists in their organization. Really, networking is the key, and it's important for empowering chemists and the scientific community in general. We really need to encourage women at all different career levels to come together and support and mentor each other.
So this leads naturally to excellent opportunities for young people, particularly women, to contribute to the work of their own professional societies and generally to the scientific and community work undertaken, of course. So all of this, hopefully good quality coffee and baked goodies for breakfast as well. Let's now hear from our friends from around the globe who want to join in and celebrate with us the IUPAC Global Women's Breakfast of 2021. Enjoy, everybody.
Salam sejahtera dan selamat datang ke Global Women Breakfast 2021 dari Malaysia.
Hello and welcome to the Global Women's Breakfast 2021 from Christchurch, New Zealand. Cheers.
สวัสดีค่ะสุภวรรณ กันตยานนท์จากประเทศไทยยินดีต้อนรับเข้าสู่งาน The Global Women Breakfast 2021 ค่ะ
日本からこんにちは。グローバル女性朝食会2021年へようこそ。私たちもいただきます。
大家好，
欢迎大家来参加由 Singapore 主办的2021年
国际女性早餐会。
Hola! नमस्कार भारत से ग्लोबल वुमेन्स ब्रेकफास्ट 2021 में आपका स्वागत है.
Всем привет от российских женщин-химиков из заснеженной России.
أهلا بكم صباح الخير من مصر.
أهلا بكم في الفطار الجماعي Global Women Breakfast.
Merhaba, Global Women Breakfast 2021'e hoş geldiniz. İstanbul, Türkiye'den bütün bilim kadınlarına sevgilerimle.
káàbọ̀ sórílẹ̀-èdè wa Global Women's Breakfast 2021.
Γεια σας. Καλώς ήρθατε στο Global Women's Breakfast 2021 από την Ελλάδα.
Hallo und willkommen zum Global Women's Breakfast 2021 aus der Schweiz.
Buongiorno e benvenuti alla Global Women's Breakfast edizione 2021 Empowering Diversity Science che si terranno nel febbraio presso il Federico II.
Hello and welcome to the Global Women's Breakfast of 2021.
Bem-vindos ao Global Women's Breakfast 2021 em Portugal e saboreiem comigo esta chávena de café. Tão bom!
Bonjour et bienvenue au Global Women's Breakfast in France.
Hello, welcome to the Global Women's Breakfast from Newfoundland, Canada.
Hola a todos, bienvenidos. Anímense, vamos a desayunar.
صباح الخير للجميع وين ما كنتوا أهلا وسهلا فيكم تفضلوا معنا على الفطور صباح الخير.
Bom dia, sejam bem-vindas ao Global Women Breakfast 2021 em São Paulo, Brasil.
Hola, bienvenidos al Desayuno Global de Mujeres 2021 en México.
Thank you very much for that. So, in our own language, magandang umaga, maligayang pagdating sa IUPAC Global Women's Breakfast 2021. I would like to officially start this event, and without further ado, may I call on my co-host, Dr. Doralyn Dalisay, to introduce our first speaker. Doralyn?
So thank you very much, Gladys. So now I'm going to present to everyone our first speaker for the IUPAC Global Women's Breakfast 2021. Our first speaker is Dr. Jessie Lepi Concepcion. She is the current president of the Philippine American Academy of Science and Engineering. She's the professor emeritus of the Marine Science Institute of the University of the Philippines, where she has led marine drug discovery and biomedical research programs and published various and several papers on subject in international scientific journals. She is an academician of the Philippine National Academy of Science and Technology and a fellow of the World Academy of Sciences. Dr. Concepcion served as the Vice President for Academic Affairs of the University of the Philippines system from 2011 to 2017. She is the founder and co-editor of the online open access journal, Philippine Science Letters. She is the editor of 4 volumes of books titled Science Philippines, which are compilations of popular science articles published in a weekly column, Star Science, which she hosted for many years in the newspaper daily, The Philippine Star. For decades, she has been a staunch advocate of the science and technology human capital development from the early childhood years of STEM education to secondary, tertiary, and postgraduate levels. Ladies and gentlemen, let's all welcome Dr. Jessie Lepi Concepcion.
Thank you, Doralyn, for that kind introduction. Here's my mug of coffee.
Thank
you. Know that, caffeine drives science worldwide. So today it's a great day, and I'm happy to be with you. And I thank Gladys, Cheryl, and Doralyn for inviting me. And I'd like to now share my screen and tell you about some of the work we do in our research group. It's now led by my younger colleagues. And at the onset, I'd like to just say that this is driven not just by caffeine, but by collaborations and cooperation among my colleagues at the Marine Science Institute, with great contributions from our research assistants, postgraduate students, as well as our collaborators abroad. And here I'll talk about work with my collaborators from Japan, Thailand, and many from the United States. Okay. And also, while we're focusing on the science of marine natural products, I like to say that engineering is so important. So engineering here is used in a broad sense. And I will show you examples of how we are doing value engineering at the aquaculture level, at the bioreactor level for our microbes, and also value engineering at the molecular level. And well, permit me to bring you on an exciting roller coaster ride. There isn't enough time to talk about all the work in detail, so I'm just going to try to give you an overview. It's going to be quite fast. And well, we're going to try to look at our research in about 5 marine ecosystems. So please bear with me. So these are the sea animals that we study: sponges and mollusks. Sponges, and here at the center are the mollusks. So you have the gastropods. They are the well-known conoideans. And here we have coned snails, and we have turids. And here at the bottom, we also have mollusks called shipworms. Even if they look like worms, they're actually bivalves. And the reason we study these animals is because we think that they present a unique ecological and biological niche where we can find bioactive compounds. Together with their associated bacteria, they provide an eco-rationale for finding small and medium-sized molecules as drug leads for cancer, infections, and neurological conditions. And that's because we have in these animals, okay, I'm going to turn off this gallery, highly evolved interactions among organisms, chemical warfare between predator and prey, and for those bacteria in sponges and mollusks, we would imagine a balancing of competitive and cooperative interactions among macro and microorganisms, leading to biochemodiversity. And no better place to study this than in Philippine waters, which is the center of global marine biodiversity in the Coral Triangle. So here we have some examples of compounds we've studied, and I'll talk a bit about them later. And this is the Marine Natural Products Lab logo in the Marine Science Institute. So that's the supply side from nature, and here's the demand side. So drug pipeline is running dry, as we know very well today. So many diseases are actually even more neglected in this time of COVID. So we need drugs for cancer, infections like microbial, bacterial, viral, protozoal infections, as well as for the aging population, drugs for pain and neurodegeneration. And there was like this decade where the focus was on combinatorial chemistry libraries. They were mostly peptidic as basis. But now there's a move to go back to the biodiversity libraries. And together with in silico analysis, we have a very powerful way of discovering drugs today and developing them at a faster pace. So let's start with the first marine ecosystem, which is the coral reef, where we have this balancing I was telling you about. And it's a very intimate environment. And you might say that it is a bit nutrient challenged. And so here we have thriving sponges and other coral reef macroorganisms and their associated bacteria. And let's just say that small is really big. So you don't see them, they're there, but they're ubiquitous and they're very important in this environment. And so now we know that there are many compounds that were thought to be a sponge in origin, but are actually being produced by their bacterial symbionts. And some of you might be familiar with this cartoon, which comes from a paper of Hanahan and Weinberg. And I understand their third edition of the book is coming out. But here is telling you that the cell is like an electric circuit with many different connected pathways, interconnected pathways. And in cancer, these circuits go awry, and there are many proteins that get mutated. And so through the years, we've been trying to study this pathway. And my PhD student graduate, Miguel, and I wrote a review. And here we have some of the compounds that we had been studying with a group of Chris Ireland of the University of Utah under a National Cancer Institute project that ran for about 10 years. And we are showing here some marine compounds that are targeting different circuits in a cancer cell. Okay. So now this Miguel is very interested in ecology. And so he did his dissertation partly on an ecological problem, which is that this sponge, Calispongius amurensis, overgrows a hard coral, Parietis cylindrica. And so together with my colleague in MSI, an ecologist, Helen Yap, we set out to do an experiment in the tank to show that there are extracts and compounds from the sponge that induced bleaching in corals and were cytotoxic to this human cancer cell line. And so this is work in progress, and we're hoping that we will be able to characterize the compound that is responsible for this potential anti-cancer activity based on its effect on the coral. Okay, so this is just a photo from a news article that came out in 2019, where a Pinay, a Filipino woman, is shown story is about how we're just discovering compounds with cures for cancer from the depths of the sea. And well, its discovery, it's a long way to a development and a drug that would be in the market. But in 2019, we published this paper with a group from my lab and the Institute of Biology, and also with our collaborators from Japan, Professor Naiko Saito, and with him, our collaborator from Chulalongkorn University, Kanitsu and Barry Brooks. And it is to show that when this marine sponge compound, reneramycin, is combined with doxorubicin, then we see synergistic cytotoxicity to MCF7 breast cancer cells, which is representative of the most common type of breast cancer, the ER or estrogen receptor positive type of breast cancer. So this is the structure of reneramycin. Some of you might be aware of this, and this is doxorubicin. And this is Jortantun, who's now a PhD student in the United States. And this is a blue sponge from which we isolated the compound. And here we're seeing the results of a study on cell impedance. And we're showing that when they're combined, then you have very, very reduced cell impedance or cell survival. And in the next slides, we're showing that, well, reneramycin M is actually 60-fold more potent than doxorubicin. So there's really a potential way of reducing the dose of this cytotoxic, cardiotoxic drug widely used in the Philippines because it is inexpensive by combining it with reneramycin. So our task was to look for the right dose combinations, and these are what you call isobologram representations of the combinations, and a combination index here that is less than 1 shows that it's synergistic. Excuse me. So here's the same slide from RTCA, and here's just a blow up of the treatment phase. But here we're showing some of our microscopic studies, and it's got an apoptotic mechanism and shown by flow cytometry to accelerate the exit from G to M phase. Right, so what else are we doing about this? Because this is something that we want to pursue eventually with Professor Naiko Saito. We've done some preliminary studies on mice, and we've done different combinations. First, we had to optimize our mouse study, and we did this by injecting it with this 41 cell line, which is a very fast-growing breast mammary mouse tumor. And so we had to slow down the growth. It's a good model for metastatic human breast cancer stage 4. And here at the bottom, we see some of our results. And I will just tell you that the combination shows a much slower growth of the tumor. Okay. So what have we done to try to secure the sponge source of this compound? So my colleagues at the Marine Science Institute have been working on a sponge aquaculture, and they've been doing it at 2 sites in the Philippines. So I think this is important for us to be able to continue the studies in vivo and eventually for bigger studies, because it's a little difficult to synthesize reneramycin, although Professor Naito's lab Nycocytus lab is focused on producing a lot of analogs of Veneramycin M, and that's actually a stabilized analog of the natural compound, and it's in the literature. All of this is published in the literature. So let me now move on to another collaborative project now with scientists in the United States. It's called the Philippine Mollusk Symbiont Icbg program, and it involves 5 institutions. And these are our collaborators: Margot Haygood, Eric Schmitz, Baldomero Oliveira, myself. They are from the University of Utah, and this originated because Toto Oliveira and I are collaborating already on another project. And then we linked up with Dan Distel of Northeastern University Ocean Genome Legacy and Gary Rosenberg, then with the museum in Philadelphia. He's now with Drexel University. And we came up with this program because there were some studies that we had published together of Toto, showing that there are microbes in cone snails. Then Margot and Eric were already studying shipworms with Dan. And so we put the group together. Toto Oliveira did this for us. And this is now ended after 10 years, and it's produced about 65 publications. And here's our documented bioresource. And we've had surveyed about 116 collection sites. We have about 820 mollusk specimens, 44 families of mollusks, 3,539 samples processed, and about 4,000 of these bacterial strains isolated, many of them bioactive and prioritized. And these are collected from various parts of our country. So let me go now to marine ecosystem 2, which is a splash zone. And that's different from the coral reef. Now, a splash zone is very, very challenged. It's nutrient challenged, it's temperature challenged, it's light challenged. So you can imagine that organisms that live here are very, very protected chemically with an arsenal. And here we study a shipworm. No, this is not a shipworm. This is a gastropod. So this is Truncatella, a very, very versatile gastropod producing a lot of compounds from their bacterial symbionts. And so this is the publication in 2020, very recent. And the first 3 are first authors, and Joshua Torres and Jertan Tun are my students and RAs in the lab, and Bailey Miller was our postdoc who'd spent time in my lab aside from time in Margot's lab. And these are all my RAs. And as you can see, it is a collaborative, cooperative effort. The thing here is we are able to find a compound that, again, synergizes with a compound that is now retired from the antibiotic armamentarium, that's oxacillin. And so there's a way for us to bring it back to become a good drug. And well, you see, synergy is something that I believe in as a drug therapy strategy, and it's how nature works, actually. So this one is active against methicillin-resistant Staph aureus, but not only that, it's also good for Enterococcus faecium, okay, where it shows a good MIC range. Okay, so this is our compound. It's a published compound, but then there was something wrong with its structure. So in this paper, we talked about, determining the absolute configuration at these positions. So I think it's also good from the chemical structure viewpoint, and also for its significant antimicrobial activity. And here we have our combination studies. So individually, they are active at very high doses or higher doses, but then when you do a combination, then you're able to reduce the concentrations log-fold, okay? And the thing about this grisharodin is it's not cytotoxic to MDCK normal cells. So that is your mammalian canine kidney cell line. Okay, now if we did further, and we did what you call a time kill study. And the blue line tells you that over time, you have the combination at that right dose, still very active over 28 hours. Okay, we move now to marine ecosystem 3, the mangroves. Very, very important ecosystem for all of us, for its protective function. And we know it is a major way for us to bring energy from the terrestrial environment, from the photosynthetic plants and trees, down to the marine environment, which is generally nutrient challenged. And a major, major player in this would be your shipworms, because shipworms are what you call the termites of the sea. They have in them bacteria that are cellulitic. So that's their main function. So this is an example of a wood-eating and wood-boring shipworm. This is a typical shipworm, and it's called shipworm because, centuries ago, they were the ones that, well, ate the wood in the ships, and they were responsible for the defeat of the Spanish Armada, okay, which was ships that were shipworm infested against the British fleet. Okay, so this is Lairodus pedicelatus, and it's wood boring, but that's not the function that we are interested in here. But I know some of our other speakers and our hosts are interested in compounds that are glycosylated. Okay, so we can look at this for the enzymes as well, but here I'm talking about the shipworms for the small molecules that their bacteria produce. So Lairodus has this resident shipworm bacterium, which is a very versatile one. It's richer in its secondary metabolite BGCs, richer than Streptomyces secolor, which is our great grand standard for producing so many secondary metabolites. Okay, and here we have Dicyathophore mannei. It's also wood boring, and look, it also has its resident Turnibacter turneri, cellulase producers. But then I'm going to show you later in another marine ecosystem, this huge, humongous, monstrous Cuphus polycalamus, which houses or has as its main resident Symbiont, a sulfur oxidizing bacterium, which we discovered and cultured for the very first time, and we named it Thiosochus pteridinicola. Okay, so the first publications on a major compound from Pteridinobacter turneri is in 2012, and here we have the boronated tartalones. And boronated tartalone E in particular is very, very promising as an antimicrobial, antibacterial, but also now as an antimalarial. Okay, and very, very recently in December 2020, we published this paper on modified oxylipins, which inhibit biofilm formation, and it's also from T. turneri. So it's a very versatile bacterium. And here we have as our main author, Noel Lacerna, who did his master's thesis with us and is now a PhD student also in the United States. And we would like to show the importance of the oxylipins, because here we have some SAR studies. So in compound 1, which is the new compound that was reported in the paper, this is named turneroic acid for the T. turneri. We have this biofilm inhibition and a very, very low cytotoxic effect on the planktonic epidermidis. So this is your staph epidermidis, which is your model for biofilm inhibition studies. We know it is a biofilm former in catheters or in the clinic. So aside from Staph aureus, okay. So here you see a selectivity for biofilm inhibition, but so does this compound, compound 6, which is 16 and 128 respectively for biofilm and for planktonic. And now we can take a look at their structures, and obviously we see that the critical functions would be surrounding the epoxide and the hydroxyl function. Okay, so now we like to look at maybe how this might be working in nature. So there are such antibiotic combinations in nature that inhibit multiple points in bacterial biofilm development for whatever purpose, for the benefit of the macroorganism, in this case a shipworm, that, that has these bacteria in the gills. Okay, so they're found in the gills and they're producing these small molecules. And what might these antibiotics acting in different ways on the planktonic and then on the biofilm phases, what might they be doing? And we are hypothesizing that they might be protecting whatever bacteria are producing cellulases, the same ones that are found in the cecum, okay, where the cellulose pieces, the wood pieces, are digested. So here we have, we hypothesize that the tartalones and oxalipins act synergistically at different phases of a bacterial life cycle. Okay, there's another major compound found in ternary, and this is ternarbactin, which is a siderophore, meaning it actually transports iron inside, and now it is being considered as a potential drug delivery agent or a cellular Trojan horse. Okay, and then now we move to the next marine ecosystem 4, which is a eutrophic marine habitat. It's sulfur rich. It is sulfidic marine lake. And the first person who pointed us to this lake was a popular newscaster in the Philippines, Jessica Soho. Kapuso mo, meaning with you in my heart. Okay, so this is Jessica, and she's holding a humongous shell, calcareous shell of a humongous or monstrous shipworm known as Cuphus polythalamius. And this is what, well, first brought us to the news, and there were several other news reports of our work. And this is giant shipworm, okay? Giant tamilok. And this is our Cuphus, and this is the shell that, houses it and protects it. And then we can compare it later with the Lyrodosis, which is very tiny compared to it, but looks kind of similar. So it is the only non-wood boring shipworm. It burrows on sulfidic marine sediments, not wood. And we don't know what it eats. And does it have symbionts? Also, it's very rare. It's the first time that a live Cuphus has been shown to the world. So what are our major findings in a paper that was published in PNAS in 2017? It is that it does not partner significantly with the cellulitic symbionts, unlike its relatives. Although we found it in the metagenome, ternary is not significant. Instead, it relies on endosymbiotic bacteria that use hydrogen sulfide to fix carbon dioxide into a biomass that can be utilized by it. And we were able to cultivate this sulfur oxidizing endosymbiote because I had this marvelous RA in my lab, marvelous Marvin Altamia, who's able to do it. And he's now in the United States continuing to work on the bacteria and the enzymes that are responsible for the life of the host. So let me just say that there's just so much more to be discovered. And this is Marvin, and this is the bacterium that we isolated from and grew from Cuphus, Thiasocius terradinicola. And what's important for us today is to know what else it is that this Cuphus bacterium with its bacteria produces. And so Noel Lacerna, the same guy who worked on the oxalipins, discovered this new compound, mindapirose A to C, and it's Minda for Mindanao, and it's actually produced by a Pseudomonas aeruginosa, isolated from your kufus. And here you have your compounds, and the mother compound is pyroluterin, and that's been published. But then Mindaperos, they're new, and among them, it's B that we're looking into, because look at their MICs, they're very low. It's really hard to find sub-micromolar MICs for antimicrobial activity. So this is considered quite low. And what's significant is it's active in gram-negative Pseudomonas aeruginosa. And that's so important because it's so hard to find such compounds. Okay, and also we see that there is selectivity, and it's not cytotoxic to MPCK mammalian cells. Okay, so this is what we use to screen, and many of you now use this escape pathogens as your pathogens, and this is our biomap screen, just to show you that we tested them in many other bacterial pathogens. Okay, so what are the discoveries of the kufus? Relatives, more shipworms discovered, and so we again made it to the news because after a century we were able to discover this new shipworm, which is anatomically divergent from the rest of the wood-boring bivalve, itself wood-boring. And so this is Tamilocus, that is the Filipino name of shipworm, Tamiloc, and you know it's edible. And we eat it in different parts of the country, like Infanta Quezon, of course in the Visayas, and also in Palawan. They're delicacies, and they're almost like oysters. Okay, and I will show you in the next slide what they look like in another place. And I'm saying that we're trying to document and protect our biodiversity. It is very unique biodiversity. So we have to do the studies on the macroorganisms themselves. And so now we're doing like what you call a computer tomography. That's almost the same as your a CT scan that we do for humans in the hospitals. So we have their morphology and their anatomy. Okay, and then of course we're also doing the genetics. Now look at this latest discovery of our shipworm, and this time it's not wood boring, wood eating. It's not sulfidic, found in a sulfidic marine environment. It's a rock boring, rock ingesting freshwater bivalve. It's just telling you there's just so much undiscovered marine biodiversity out there. And that's there. Like, you know, we're hammering the wood and getting out the white, very, very white, very translucent, bulby, cute shipworm. And we've called it a new name. It's called Abatania. Okay, it's an Abatania. Lithoredo, because it's rock boring. Abatania, because we found it in the Abatan River in Bohol. Okay, and it actually looks very, very edible. Okay, so we're saying this, we're showing you the mollusk because each mollusk is a unique biological and biochemical niche. It's got a unique habitat in itself, living in a unique ecological habitat, and so it is a potential source of a unique microbiome and a unique chemiome. Each of those microbes in the microbiome has its own chemiome. All right, so we're now done with 4 marine ecosystems. We're off to the last one in this roller coaster ride. I don't know how many more minutes I have, but I think this is very important. This is funded by our Philippine Department of Science and Technology, but it's also dovetailed with the Icbg program, which is funded by the US NIH, the Philippine Mollusk Symbiont program, and my collaboration with Baldomero Oliveira of the University of Utah. And this is Pharmacies, original name, and it's now called the Drug Discovery and Health Program, Marine Component. And many of you are familiar with this. So many of you know of the conoideans that are found living in sediment near coral reefs. And this is the work of Baldomero Oliveira and his team for decades. And in his work, he focuses a lot on the ecology, okay, on the behavior of the cone snails relative to their prey. And so there are many strategies here. And he says there are cabals of venom peptides. So these are venomous snails that are full of peptides with strategies to capture the prey. So these are the different strategies. And there are now other more updated taser and tether strategies that Toto Oliveira uses. Okay, and what it has is a venom duct that delivers the peptide through spears. So there alone, you already have the very, very early drug delivery system discovered by the snail 60 million years and greater, long time ago. Okay, so see, here are some of the peptides that are well known in the literature, and one of them is actually a drug for intractable pain in cancer. And in the Philippines, this work was really led by Baldomero Oliveira and Lourdes Cruz. Now, what's the challenge for us, and what's the opportunity for us in Asia? Well, I'd say let's try to develop a conoid drug. So right now there is Prialt or ziconotide, and it was a shaker peptide that is in its original sequence and 3D structure, that is the drug called Prialt. They tried to change every amino acid. It's not as good as the one that nature produced. And now we have others in the lab and with Dr. Oliveira that he would like to pursue as drugs. And in particular, we have Contulakin G. That is a Filipino name, meaning pushing effect, sluggish peptide. It's got to be pushed, okay? It's a neurotensin analog and a neurotensin receptor agonist. So it's acting on GPCRs, okay? So it's hormone-like. And then we have Conantukin, antukin in Filipino means sleepy. So here we have a sleeper peptide, and it is an NMDA competitive antagonist that is selective. We have this in clinical trials, acting on alpha-9, alpha-10 nicotinic acetylcholine receptors, and more recent discoveries, we have coninsulin. Okay, so it turns out Conus geographis also produces insulin, but in a truncated form, and so it's very, very fast acting for prey capture. So it gets the fish hypoglycemic. And more recently discovered by my RA, research assistant and thesis student, Bea Iris Ramira, is consomatin from Conus rolani, also truncated compared with somatostatin. It is the first to target a somatostatin receptor subtype. So, conoid peptide drug developed by an Asian consortium as an alternative to opioid drugs, where you know there is an opioid epidemic in the world. This is for pain, for us, for an aging population. So there are many open questions. So now this is going to be very, very fast speed roller coaster. There's publications on con insulin, and here's the impressive conoidean biodiversity. We're barely scratching the surface. So we're looking at cone snails, but there is the turids, 12,000. This is 12,000 species, and the taxonomy is very, very problematic. So we're also working on the taxonomy. And here we have a way to access these conoidean venoms that are from the engineer fishermen in our islands. This is in Bohol, so very close to where Dora Lynn lives. And so we learn a lot from our fishermen, and we consider them partners. And by this method called lumen that they devised, so these are like old tangled nets, and we leave them down in the sea, and then after some time, we get an ecological harvest. And there's so much richness in this collection from the lumen nets. You'll see they're all conoidean morphospecies. Many of them are never before studied or even documented in our history. Okay, so now here's a summary of our Philippine DOST funded project, and we have many cone snails here. And I will just tell you a bit about one from the Turids, which is even less well studied. They're the cousins of the cone snails, and they're much older than them, and they're harder to study because they live in deeper waters than cone snails. And so my graduate student, now also a PhD student in the United States, Victor Chua, discovered this drilipeptide, which is from Clavus David Gilmore. And you will wonder why we call it David Gilmore. It's because of its phenotype in mice. It produces this foot stomping, paw foot stomping or foot stomping. And so it was named after this rock singer. Okay, so this is Victor, and it's just showing you how complex the venom, the crude venom is of our Turids, just like the cone snails. And many of you already know that there is like a structural framework for the cone snails, and the Turids follow the same types of structural frameworks. Okay, so here's the bioactivity that we discovered: shaking, thumping of the paws, frequent scratching and grooming, and seizure when injected into young mice. But then we did further. We isolated dorsal root ganglion neurons from mice, did a primary culture of them, and did what we call constellation pharmacology, where we're able to show through calcium imaging that our compound is active in subsets of these neurons. So they are different types of neurons. It's a heterogeneous population. And some of them are the same neurons that respond to certain agonists. So this is what we call molecular target-based, cell-based screening of our compounds. Very, very powerful assay. Okay, so what else happened with the cone snails? And this is, I promise, the very last part of it. Sometime in 2009, we were able to discover so many bacteria in each of these cone snails. And that really became the basis of our Icbg Philippine Mollusk Symbiont program. And we were able to get the funding from the US NIH and Fogarty International. And so we've continued the work. And here are some of the results. So we published on the polycatins from Conus polycarius. So these are thiazolin metabolites, and they are active against this subtype of serotonin receptor. And no amount of modification of this most active compound, polycatin B, polycatin from polycarius, no amount of changes on the left side or the right side of it gave activity better than the native polycatin. It's just telling you that we have our models from nature. Here's another example. So this is a peptide, but not a ribosomal peptide. It's an NRPS peptide, non-ribosomal peptide product. And this is called nobilamides from Shicorius nobilis. And so we were able to isolate these compounds and show that they're active in TRPV1 receptors. So many of you may know that this is also found in your DRG neurons. And so they are also active in your DRG assay, in particular to those DRG neurons that respond to capsaicin, which we know is a anti-pain agent. It's actually found in a lot of products already in the market, including Salompas. Okay, this guy, Oliver Belleza, so he is a master student of my collaborator in Institute of Chemistry, Aaron Villarraza. So he did modifications of the bilamides and also synthesized them. And well, these are the results. So we've also filed IP for this. So Oliver is doing his PhD in Europe. All right, again, each mollusk has its unique habitat and therefore is a potential source of a unique microbiome and a unique chemiome, and therefore we have to study them as a source of new drugs that we need for the human population. And we need all of these types of studies, so we must learn to do our science well. And not only that, we have to understand the molecular targets, so more and more science, more deep science. And so that's the only way that we as humans can look at our terrestrial and marine biochemodiversity as a source of drugs and nutraceuticals. And the best models for drugs are derived from nature, and humans can learn important lessons on creating successful ecosystem societies from evolutionary biology. The survival and success of humans and the rest of nature are closely intertwined and must be pursued holistically. And that is why we need to conserve and protect our biodiversity. So this is my research group in one of our science retreats. And here we're having some fun, but I also asked them to prepare small posters on their range of work in my lab. And here we were surrounded by this beautiful trumpet flower, which could actually be deadly because it produces hallucinogenic compounds. Okay, so beautiful and deadly. That's what we have in nature, both terrestrial and marine. And so I'd like to thank you all for listening. I'm sorry this took long. And they're in their PhDs, these guys. They're all doing their PhDs, 1, 2, 3, 4. This is Bailey, the postdoc. Jessa is still doing her master's, still in the lab now with Lilibet Salvador Reyes, who is the leader in MSI. And this guy, he went to medicine. Okay, he went to medicine. And this girl, she's resting. Okay, she's a molecular biologist. So thank you so much again. As I thank my collaborators and funding agencies, I'd like to thank you for your kind attention. Thank you.
So thank you very much, Dr. Concepcion, for presenting a very comprehensive report and science outputs from your lab, highlighting the importance of marine ecosystem as a source of bioactive compounds, whether small molecule or big molecule, that could target diseases that are important in our society. So in interest of time, we will have 2 questions from the participants. So we have here the first question. As a woman leader in science in the Philippines, what are the challenges and how did you overcome the challenges to be one of the successful woman leader of science in the Philippines?
Thank you so much for that question. Indeed, very, very challenging because all of us, we want to be, accomplished all around. And in Philippine society, our first role is to be a mother, a wife, right? To stay at home. So I did those. But then in our society, we also have extended family. So we have them taking care of our kids. And as long as we have developed this passion, this interest to pursue our awe and wonder of nature, then we can do it. It's really a lot of determination, self-determination, and belief in oneself, and belief in science as the highest form, to me, of human activity, rational and also emotional activity. And, you have to be prescient before scientific. You must be sensitive. You must be interested in your environment. Well, not just in your family, not just in your children, but also in the world around you. And then you must have developed the discipline through the young years in youth and childhood to work hard and to be rigorous in your work, to excel in school. And that is really, the challenge now, Doralyn, that I'm asking you to join me in addressing in the Visayas, and that is STEM education. Starting at the secondary level, we're thinking that the junior level is where our high school students will decide to go to STEM, the STEM track in K11-12. And now we have to connect them with the leading state universities and college and private higher education institutions in the regions. We need the champions like you and Janelle, okay, to try to link up with your high schools and try to get those young women in particular to get their careers going in science. It's very tough.
All
Very
right.
tough.
So thank you very much, Ma'am Giselle. So another question. We have young faculty members here, young women faculty members.
Yes.
What advice can you give them on choosing a niche? So your niche when you created your own program was a marine ecosystem. And there are some other niches that they could develop into. So what advice can you give to our young women faculty, budding scientists on choosing a niche to create their own research program?
Okay, I'd like to address the young women, especially in our regions. Okay, those in the SUCs and the private HEIs. And so, as part of PASE, we want to go outreach to the regions. We recently came up with PASE S&T Human Capital Development paper. And there you will see that for you, the PhDs and the researchers in your SUCs, we want you to look at the problems in your region and the opportunities, challenges to develop your region. I know that's like applied science right away, but there's just so many things that we could be interested in, depending if you're a chemist or a biologist or an analytical chemist or a computational chemist. It really depends on resources provided to you by your SUC, but bottom line is follow your dream, pursue your passion, pursue your personal interest in your field, because surely you will be able to connect that with something in the region that needs attention. So we say, come together as a group, try to recruit little groups of yourselves, among yourselves, and collaborate, cooperate, not just within your SUC, but among our scientists in universities, leading universities in your region, and then adopt others, okay, the second tier universities, you might say. And remember to collaborate. Remember to collaborate not just among yourselves, but with foreign collaborators as well. And enrich your teaching to benefit the next generation with your own research. Okay, so those are some of the strategies. I think collaboration, cooperation is very, very important from my own life experience as a scientist. Okay.
Okay, thank you very much, Dr. Concepcion, for sharing your time, expertise, words of wisdom, and particularly the passion that you put into your work. So thank you very much.
I think passion, that's a very key word, just like Dora Lynn, so much passion. Yeah, it's driving us. Yeah.
Thank you. Thank you very much.
Thank you so much, everyone.
So now I will give the floor to Dr. Cheryl Aureola to introduce our next speaker. Cheryl?
Okay, thank you, Ma'am Doralyn. Good morning, everyone. Our next speaker is an alumna of the University of North Carolina at Chapel Hill, USA, where she received her PhD in biology in 1998. She became an NIH postdoctoral researcher at the University of Texas from 2000 to 2002, and a Humboldt Fellow at the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, Germany from 2003 to 2004. Currently, she is a professor of molecular biology at Suranadi University of Technology, or SUT, in Kurat, Thailand. Her research works focus on the directed evolution and phage display biotechnology, notably on enzymes for biotechnological applications and engineering of antibodies for therapeutic and diagnostic purposes. She has published more than 60 international peer-reviewed articles, filed patents, and was granted more than $8 million for her research projects and development. It was also under her supervision where students and researchers from different parts of the world joined SUT because of the university's Excellence in Biotechnology program. Ladies and gentlemen, it's my pleasure to introduce to you Dr. Montero Mon Yamabay. Dr. Mon?
Yes, thank you very much for your kind introduction, and good morning, everybody. Still enjoy your breakfast. So first of all, I would like to thank you, Cheryl, and the committees for inviting me to give such an interesting talk. My talk today would be slightly different from the previous talk with a lot of science. I'm going to be more into journey and tell you story of my work. I will be less science and more of the story. But of course, you could ask me more in detail later. Okay, so I will share my screen now. Okay. All right, so, oops, this is not what I want. Wait a minute. Let me go back, and I have to share it from here. Okay, let me try one more time. Okay, so my slide should start from this one. All right, so the title of my talk is My Directed Evolution, and probably later on you will know why the title is like this. So, and again, this is the first time I know about this. So I'm going to tell you a story of my evolution. I guess the committee told me that maybe I should talk about my journey. Of course, my journey started 54 years ago. I entered a school, Chulalongkorn Demonstration School in Bangkok, Thailand. So the first turn in my life is that I entered a faculty of pharmacy in Mahidol University. So that trained me to be a pharmacist. And after that, I received a scholarship from the Thai government to work for Suvarnabhumi University of Technology, but the government sent me to do my PhD in molecular biology in UNC Chapel Hill that Cheryl have mentioned. So, and then that's one of the big turn for me. And then I think the other important point in my life is that when I did my 2 postdoc in molecular cell biology at the University of Texas Southwestern Medical Center, and also next to the Max Planck in Dresden. And then I'll become staff of a School of Biotechnology in Suvarnabhumi. I've been there for almost 20 years. I never moved to any other place. I built myself up from just a faculty member to a professor right here. So I'd like to tell you my story about my establishment of my work here. I guess with the beginning, so I started, you have born because of the parents. I was born in a family of, my dad is a professor in speech communications, and my mom is a teach mathematics. So I grew up in an academic background. But then if you think that they forced me to study, and it's actually not the case. So I'm pretty a mediocre student when I was a student in high school. I never get good grades. I am a bit insecure in myself. I love to play sport. The only thing I'm excel is doing sport. But I have to say that I have a very wonderful science teacher, even though my grade is not good, but I still remember one time this teacher asked me to do some experiment, and I think it's not very good, but she did said, hey, Montero, you have a good thinking, you can be a good scientist. I think that what she said is always in the back of my head, and I said, okay, I'm not that bad. I could be a good scientist. And I think that keep confidence, keep pushing me until today, even though I was not very good then, but I believe because she said so. So the teachers in elementary school and high school, I think they are very important. The other important teacher is my English teacher, and he is also a classroom teacher. These are current picture. I'm still in contact with them. He taught English, and I think English is very important to open the door into science. And in Philippines, you use English as almost for a second language, but in Thailand, we don't have English. So if you are good in English, it's really helped a lot. So I owe my success to these two teachers from high school. All right, and then as I said, even though I'm mediocre, I managed to enter into the pharmacy school, and I had many great teachers from pharmacy school as well. And one of the teachers always said, hey, from here, you can go anywhere. And that is the case for me. I never practiced pharmacy. I think very short time, I entered into the graduate school right away. But what pharmacy school taught me, I think it's very important is to be precise and extremely careful, because you train that is after why patient die. But that skill, I think, is very important that I use until today, and I try to pass on to students, because when you're doing molecular biology, you have to be precise. Everything is so small. And I am grateful for my training as a pharmacist there. All right, so after I finished, as I said, I never become a pharmacist. I received a scholarship from the Royal Thai government to study PhD to come back to work in SUT, so they pay me. So the next important teacher in my life is my PhD advisor, Brian Kay, who taught me the topics then is the phage display technology. It's a hot technology 20 years ago, just like CRISPR-Cas now. So he taught me phage display, but also he introduced me to how to be a scientist. Freshly graduate from pharmacy school, you don't really know about scientists, but he taught me a lot about phage display. All right, so what I did what probably if you never heard about phage display is a technique of molecular evolution. It means you evolve in a way that you look for a peptide or protein that can bind to interact with target, and that is through several rounds of selection. That's called evolution for something that binds strongly. So I did my research on the study of a protein called intersectin, and I used phage display to map protein-protein interactions in the cell, and I managed to, using this technique, I managed to predict the function of intersectin that would be important for endocytosis. And later on, they published later for my work, not Nature is not my work, but then based on that discovery. So people now know that intersectin is involved in endocytosis. So I got some training in molecular biology from that phage display. But right after I finished my PhD, I feel that I came back. Actually, I came back to Suranari for a short time, but I still feel that I'm not ready to teach. And everybody went for a postdoc. So before I came to Thailand, I met Dick Anderson. He is a cell biologist, and he works on endocytosis. He is the father of a receptor-mediated endocytosis. And I've been wanting to do cell culture, study cell biology, and I was lucky that I get to do postdoc with him on molecular cell biology, working on mechanism of EGF receptor internalization. So I did bring the knowledge from Brian Kay, which is engineering. So I engineer so many kinds of the EGF receptor precisely using a technique called sticky PCR that I developed with Brian, and we used that technique, and I get a chance to do cell culture or the classic, isolation of cell components using ultrasound diffusion, immunofluorescent data. So we managed to help understand more on the signaling of EGF receptor. The thing is that I learned this, I published this, but it's impossible when I came back to Thailand to adopt these cell biology techniques in Thailand, because all of these are using fancy equipment. Cell culture is too expensive. So, I went back to Thailand and I still couldn't do anything, but while I was working a little bit, there was an announcement for Humboldt Fellowship. So I asked Dick to write me a recommendation letter so that I could go to another postdoc in Germany in his competitor's lab. Dick discovered Caviole membrane system, and then Kai Simons in Germany, he discovered lipid raft membrane. Probably some of you have heard. So these are probably the same thing, but the two big scientists in two parts of the world name is differently. So I asked Dick, and he's kind enough to write me a letter so I could get a scholarship and work in Kai Simons lab on a similar membrane system, but they call it differently. So, but then because I already a staff at SUT and I couldn't leave any longer, even though the scholarship gave me two years, I could do only 6 months in Max Planck, which unfortunate, but it's also fortunate things because, and you know, this is the first time that I got introduced into research in Europe, into scientists in Europe, and it is just it's not the same. It's similar, but it's not the same as a culture of science in the US. So it's really opened my perspective about science. And not only that, so I didn't get any publication out because it's only 6 months, but it's introduced me to science in Europe. And it's also, some of you may have known, it's also introduced me to a lifetime collaborator from Austria. In fact, I met him already in Thailand a short time, but then everything grew when I was in Dresden, you know, and all these pretty European countries. So we fell in love, okay? And then I was brave to got married to him because I have to work in SUT. Maybe this is a more personal, but I like to tell you that at that time, I don't want to worry about future, okay? I'm just, he is still in Austria, I'm in Korat. And we decided to have a long distance relationship. And they said, okay, gonna be 3 year, 5 year, and we do something. And you know, it's been 16 years. We keep long distance relationship and we keep long distance collaboration. So I think I could tell you that if you question long distance relationship, I said it is possible. And I think it's because you have these collaborations as well. So because of that, you know, I established myself here as Suranari. And what I can do, and in that time, even though Suranari looks pretty, but we don't have that much of the equipment. So luckily, phage display technology is actually a microbiology techniques, plus the expression of things in E. coli. So when it's microbiology, it's doable, it's not that expensive, you just need an agar plate. And this is a picture of an agar, a bacteria that is infected by the phage, and the phage turn blue because you add some a marker. So I can do this. I can start to establish phage display in the lab. But the other thing is that phage display is in fact expression of protein in E. coli. And because of that, when I met Dietmar, I decided maybe I would change into not only express phage, but express enzyme, because Dietmar was working on enzymes. So I established the lab in Thailand, expressing enzymes, because that is doable in addition to phage display. So the first generation of my lab, I have a time to close supervise my student with a very limited facility, but try to establish. And because I have Dietmar, and so we collaborate, I send my student to him, he send, and we have he get me to know another scientists like this, Vincent and Peter and Brian from the US is also still supporting me. So this is how I established my lab from the technology that is possible in Thailand with the limited funding. So in the beginning, I shipped my work from, I cannot do cell biology, but I can express enzymes. So the first paper was expressing these enzymes, several enzymes in E. coli. I managed to publish in Csir Biotech. That is 20 years ago. There's no way that you can publish a work like this, just cloning enzyme expression in Csir Biotech anymore. But then that is the beginning of my work. And I keep continue working on expressing enzyme for quite a while. So if you go back, I working on phage display technology, and this is can be called that is a molecular evolution technology, because you basically select for something that has a specific function that you want. And at the same time, there was a scientist women scientist named Frances Arnold. She developed this technique called directed evolution when she used a genetic engineering approach to create a random peptide enzyme library, and then she did the selection. So she called that directed evolution to try to improve the enzyme to suit various industrial application. So I decided to adopt her techniques and do the research in my laboratory as well, because I am familiar with phages play. So is this a difficult techniques because you have to build a library, but it's possible. So we build a library. But the first PhD student who did with me, in this is her result, we have a nice result, but we picked up the wrong enzyme. I picked up amylase, and the enzyme is so good already in the market, in the industry. So it's hard to try to improve it better than what is already there. So the student managed to finish her PhD, but we didn't really publish in a nice paper. It actually worked with the second student. It's actually master student. We changed the enzyme. We tried to direct it evolve chitinase instead, and nobody worked on that chitinase as much as amylase. So this student is with a master degree. She showed that she can improve the enzymes slightly better, but we managed to publish in Biotech Journal. Again, I don't think we can publish something like this in Biotech Journal now anymore. So that is story about directed evolution. And as I said, I came back here to Thailand with this limited supply. So I think the other thing that in my head is that I've been working with phages play of a short peptide, but maybe we could do something different. So we decided, I have another PhD to join, she's pretty good. So we decided to maybe make a library of antibody on the phage. But of course, you cannot express the whole antibody, it's too big. So we can express a small fragment of antibody on phage. And because of that, we started to do this project. We collect blood from 140 healthy Thais around Korat. We extract mRNA from B cell. We build a library of the antibody and put onto the phage and display, and we create a library. So I'm proud that we can make a made in Thailand antibody library, and it's been useful. It's been useful until now, because we can collect, we can obtain many good antibody from that library. We found more than 10 patents application of the antibody from that, and we also give it to many colleagues around the world to use it through collaborations. And that was published in 2009. The directed evolution also published on 2009. And you can imagine that in 2018, you all have heard that the Nobel Prize was given to Frances Arnold, George Smith, and Gregory Winter on molecular evolution. You know, I feel almost I received the Nobel Prize myself, because I've been working with phages play, phage antibody and directed evolution, and these techniques finally recognized with the Nobel Prize. So it was really happy moment for me. And also, is this the same year that I was received a recognition as a senior fellows Thailand Research Fund as well in the same year. So that means also that I am old already. I can't believe that, all of a sudden I become white hair and everything. So that is something about my technique. So probably why I call my journey as a directed evolution. And anyway, because of DITMAR and all the network, as Gladys already said, networking is very important. I have many exchange students, more than 30 exchange students from Austria come to the lab, and that helped internationalize the lab and make my student exposed to other students, improve their English quite well as well. All right, so I think in the beginning, the work in my laboratory is more about expressing enzyme. We have E. coli, and also we move on into Lactobacillus plantarum, and this is through collaboration with DITMAR. So we have a secretory production system, and this is what I learned with the phages play. I can adopt that into expressing enzyme. It make it secret out, and it's very efficient. We publish a number of papers from the work on the enzyme expression then. But then this is all published work. And there is a time when you really need to get your work have more impact than just putting on the shelf. Everybody said, okay, people just publish and put on the shelf. We have to get use out of it. So I pick up there are two successful enzymes that have a future for a real application, and these are manganese and chitosanase. So we continue working on these two enzymes, and we put these enzymes into the food grade expression system, not only in E. coli, because people are not very, it's not very well accepted. So with the work with Dietmar, we produce this enzyme in food grade lactobacillus as well, and we plan to use it for a real application in the future. So let me give you a demonstration of the application of the enzyme that we've been working on for the last 15 years. And this enzyme is a chitin and chitosan degrading enzymes. So just like in Philippines, we have lots of factory, crab, prawn, and we have waste of this shell. And so in the process of this, people produce chitin and chitosan. And in my research, I would like to use hydrolytic enzymes of chitinase and chitosanase to generate chitooligosaccharides. Of course, in the future, we could try to replace all the chemical reaction through using enzyme technology. But at this point, I only focus on using enzyme to degrade chitosan into chitooligosaccharides into a well-defined structure. To do this, I have a colleague from Norway to have this expensive machine so that he can help analyze the chitooligosaccharides products from our research. This is a bottleneck of the chitin chitosan because the product is so heterogeneous and is not well-defined. So I decided to work on a well-defined product too, so that when we have we want to apply it so we know exactly what it is. So Vincent Ising from Norway helped me identify, analyze this product well. And just last week, we managed to publish for the first time that this well-defined chitooligosaccharides indeed has an anti-inflammatory activity. And the good thing is that our chitooligosaccharides is soluble. So this is my next direction. We would like to, Thailand also tell, okay, you have to create something useful for the country. So I'm thinking of making a cosmeceutical and nutraceutical innovation out of this product. So we are, the direction is we move toward this part now. And also, okay, when we come back to the antibody phage display, I've been having students come and isolate antibodies against various interesting targets that we managed to file, as I said, more than, I think last time we checked about 15 patents on this antibody fragment. And, as probably people have, that phage display, antibody derived from phage display are already in the market by FDA. The problem is that the price of the antibody drug is so expensive, and it's predicted the value will increase to 300 billions in 2000, but everything is still expensive for Thai and also the Philippines and our country. I hope that my work can help reduce the price of cancer drugs so that everybody can get access to. So this is my wish, my goal in the future, that we can produce antibody drug in a much cheaper price. So right now, if Gessler said about this passion, I think everybody has to have passion, and I think I have my passion and my motivation. I want to do some product. For one part is for the prevention of disease, and maybe it is anti-inflammatory or whatever, and especially anti-chronic inflammation, which we know that this is a cause of so many other diseases. And on the other side is a treatment, if already there. The treatment here is that I try to reduce the cost of the treatment that is very expensive. So with the phage display work, we published a number of papers already. We did antibody engineering. And of course, because we've been working with phage display, we are used to this shuffling. With the phage display, we can do the shuffling of the antibody to improve the activity. So I'm working on that as well. We start to use antibody for bioimaging because I'm from a school of Institute of Agriculture. So we use antibody to image. This is a nitrogen fixing bacteria in the nodule of plants. We published that, and we also recently a PhD student, now a postdoc, looking for antibody against Pseudomonas. This is for bioimaging, and so of course we hope that one day we could maybe generate a novel antibiotics to treat multi-drug resistant infection. And the last part of my work that I have to do, as I said, if you really want to put the product into market or to the customer or to people who need, you have to produce it in a right system. So I've been trying to establish a mammalian expression system to generate antibody, and these are all very expensive stuff. I can have some money to run this, but it's still expensive. And it's not easy because you look like you're competing with a big pharma company, but at least I would like to try to set up a platform that would be reachable for a much lower price. So this is my drive at the moment. To say we have the antibody, SCFV in the top here, but with the genetic engineering techniques through antibody, we try to build a platform now with the postdoc in the lab now, and we can make an antibody drug, and I hope that that could be useful in the future. So, and also, you know, when you want to produce drug for a long term, you have to establish a stable cell line. So I am trying to build this platform, and it's not easy. It's quite costly, but that is what we would like to do. All right, so I guess I come to the end of my talk, and you can see the picture of my current picture of the people in the lab now. Yeah, it's always fun to work in science. And right now, my student, I have students from Myanmar, from Russia, that girl from Myanmar, and from Indonesia. I never have a student from Philippines though, but anyway, I confirm that the networking collaboration is very important. And also this is just to show you the picture of my labs from the beginning until now. I have no time to mention everybody, and these are all the workforce to help me create the work until today. And yeah, so these are the pictures. On the bottom right here, this is the current laboratory that I have now. I have more funding now compared to before. And of course, this is a long time ago. Nika is taller, that almost as tall as me now. And as I said, international collaboration is important. So, you know, married to Dietmar is make me easier, I guess, to collaborate. So I have a number of collaborators from Europe, and all, but there's still some in the US and also in Australia. And all the funding, basically, you try your best to get funding from everywhere. So I basically receive funding from almost everywhere to try to run the lab. All right, so I guess that is my overview of my talk, right?
Thank you, Dr. Monty, for sharing your life and some of your researchers. It's really a privilege to know your family because for those who don't know yet, her husband is my Austrian professor. So I'm just curious, how do you balance your time as an academic researcher, as a mother of a very fine young lady, and then as a wife of a very active researcher?
Oh, okay. It's not easy, you know, but then I think I'm lucky that I live on campus. So it's close by. You know, sometimes I went home, you know, put Nika to sleep, and I used to come back to the office and work. Okay, so, and I think, as Gisela said, I think passion drive you. So if you really enjoy what you're doing, you could do it. Yeah, I guess.
Yeah. And it's really good that you have same interests. Dietmar and you have the same interests. So that's why you're really very compatible. Okay.
Right. He helps a lot. I mean, especially in the beginning with the enzymes. I came to work on enzymes because of him. Yes.
Oh yeah. And don't worry, I'll send some students to your lab once the travel restrictions is lifted.
Sure, that would be good.
I'm also curious because Thailand has so far succeeded in preventing widespread transmission of COVID-19. And as the head of School of Biotechnology in SUT, can you share some challenges you faced or you're facing, especially during this pandemic?
In terms of everything, right?
Yeah.
Yeah, of course. It is a big issue now, but Thailand has a second wave. I don't know if you heard, and that is through the borderlines. People from Myanmar come, and then I think that is when it has the second wave. But I think Thai people are afraid of COVID, so they wear masks. You know, so I think so that helps us. We manage to control everything, and of course. The good thing is that now everybody can use Zoom easily, you know, because we have to close down school. So yeah, so it actually that's a good side of COVID. And of course, hopefully we'll have our vaccine in 2 months, and that would probably help because of the economy is running very bad now. Yeah,
okay.
Right.
Okay, thank you, Dr. Monty, for sharing your time, and hope to see you soon.
All right.
Okay, thank you very much.
Yes.
Ma'am Gladys? Ma'am Gladys, Ma'am Dalisay?
So, yes, congratulations, Montero, for a wonderful, inspiring presentation. So I just have a question. Maybe it's all for everybody, especially for the students who are aspiring to be a successful scientist, just like you. So as a successful scientist in your area, what was your catalyst in doing well in science? What really initiated this? Because you mentioned at the start that you were a little bit of a mediocre student, but look at you now. You're very successful. And yeah, so what was your catalyst?
What my catalyst? And I think, okay, first of all, I think teacher. If you have good teacher, I told you about that science teacher who said, okay, you're not that stupid. You know, you can do good science. Sometimes a little bit of that, I think it helped me. And also, I'm lucky to meet the teacher, my Brian K, my PhD advisor. He is my role model. I follow him and I can see his passion and you want to be like him. I think the catalyst is that you enjoy, you have this passion and you follow footsteps of other. I think coaching is important. And the second teacher, Dick Anderson's in cell biology, I see he's passionate in science and I start to feel it that way. And when you have the passion, you're not afraid of the trouble. You just keep doing it. Of course, you have to be persevere, hardworking, but if you really enjoyed it, I think you can work hard for that. I think that's a catalyst. I think the answer would be passion is a catalyst.
Yes, I agree. You have to be really. So
okay, go ahead, Gladys.
So Doralyn, yeah, you have a question, and then after that, I would like to present the certificate of appreciation to the speakers before I run out of battery, because up to now I don't have electricity here in Upilos Baños, so I would like to present the certificate of appreciation to the speakers.
Can I have one question for Professor? Great talk. Thank you for walking us through your life experience as a scientist. So my question is, would you recommend young women in the future that they will pursue science or career in science in the future, and why?
Oh, yes, of course. Because then you become independent of men. Okay, I think it's satisfying to be a scientist, and then I really think that okay, men would just fulfill your life. Okay, you don't depend on them. I think it made me so. I think I think you are as good as men in do. I think probably you will be better. So I think you should do. And if you enjoy it though, not do it. But I think if you enjoy science, just do it, and it's doable. And I think 80% of students in my lab are women, and they are better skilled sometimes than men. So I think, sure, please do it if you like it. Okay.
Okay, thank you very much. Gladys?
Okay, thank you. So before we go to the last speaker, I would like to present this certificate of appreciation to our guest speakers. So let me just share. Okay, so yes, so and I would like to read it. IUPAC Global Women's Breakfast 2021 and the Philippine American Academy of Science and Engineering award this certificate of appreciation to Gisela P. Concepcion for sharing her presentation entitled Learn and Do Science. Learn and do science to save our biodiversity and discover its treasures at the IUPAC Global Women's Breakfast 2021 with the theme Empowering Diversity in Science held on February 9, 2021. And this was signed by yours truly, Gladys Completo, as the organizer and the host of this webinar. So Giselle, thank you very much for accepting our invitation. So this is your certificate, and then I will send it to you virtually also.
Thank you. Great honor and pleasure. Gladys, Cheryl, Doralyn, Montero, you're all inspiring. I always say I get my inspiration from the young as well. Of course, from my mentors, but also from you. Thank you.
Yes, thank you. And then next, I would like to give this certificate of appreciation to Montero Yamabay. Thank you very much for joining us and for sharing your journey, your directed evolution at this IUPAC Global Women's Breakfast 2021. And we look forward to collaborating with you now that we know that you're working on, and maybe you can share your experience and your lab techniques to our students as well. So this is signed by our PAASE President, Ma'am Gisela Concepcion, and by myself. Thank you, Montero.
Yes, thank you very much. It's a very nice conference. I'm glad I get to meet new friends, and of course we could collaborate in the future. Looking forward to that. Thank you for this event.
Yes, and last but not the least, okay, before she gives her talk, I would like to present this certificate of appreciation to Ramya Chakravarti. She was my previous colleague at Scripps Institute when I was a postdoc. She will be sharing a presentation entitled A Sweet Trail Mix of Glycans, Proteins, Microbes, and More at this webinar. Thank you, Ramya, for joining us. And I know it's so early. We are 2.5 hours early ahead of India, but still she accepted this invitation. Thank you, Ramya.
Thank you, Gladys. It's really nice of you to show this appreciation even before my talk. Yeah,
yes.
It's nice being here.
I would like to stop share now, and I will be introducing our next speaker, which is Dr. Ramya Chakravarti. Ramya is a principal scientist at the CSIR, Institute of Microbial Technology, Chandigarh, India. She's an active researcher in the area of glycobiology. She graduated with a PhD in molecular biophysics from the Indian Institute of Science in 2006, and subsequently trained as a postdoctoral fellow at the Scripps Research Institute, La Jolla, USA, before returning to India in 2010 to set up her independent research group. At CSIR Institute of Microbial Technology. She has initiated research on microbial glycan binding proteins and carbohydrate active enzymes. She mentored the research of several PhD students and organized many science outreach activities for school children. She lives in Chandigarh, India with her scientist husband, Dr. Shikrishna Subramanian, and their two adorable daughters. Let us all welcome Ramya. It's all yours.
Thank you so much, Gladys, for the kind introduction. Let me just share my screen. Thank you, Gladys, for inviting me here to this Global Women's Breakfast 2021, even if it's a virtual breakfast we are sharing. Gladys and I know each other from our postdoctoral days, as she mentioned, a decade ago. And even though we didn't work on the same project, I managed to pick up some chemistry from her, for which I'm grateful. We also shared lunch frequently together, and from that I realized that Philippines and India have a lot in common in terms of culture and upbringing and environment, etc. So today, when she asked me to talk about my scientific journey and research, so I came up with this title, A Sweet Trail Mix of Glycans, Proteins, Microbes, and More, which is basically capturing the research interest of our lab. I haven't really done this kind of scientific journey talk before. So, but it was also nice preparing for it because it gave me a chance to look back and see where I have come from and how I landed here. So I will try to talk a little bit about what I have picked up during this journey. And I hope there is something that resonates with at least some of you in this diverse audience. So I first like to show you where I work. This is the CSIR Institute of Microbial Technology. We are in Chandigarh in northern India at the foothills of the Himalayas. And Chandigarh is a very beautiful planned, well-planned city. This is the official emblem of Chandigarh, basically an open hand, free to receive and give peace and prosperity. This is our institute, the CSIR Institute of Microbial Technology. It is one of the labs of the Council of Scientific Industrial Research of the Government of India. And we have a very beautiful campus with beautiful gardens, which win prizes every year. And apart from this, we also have very well-equipped labs, a microbial type culture collection, which is a unique repository of different kinds of microbes, a biochemical engineering research center, which helps to take processes from the lab scale to the industrial scale, a G.N. Ramachandran Protein Center, which is involved in research in protein science, engineering, and structural biology, and also bioinformatics and big data analytics. And we also have research in infectious biology, biochemistry, molecular biology, immunology, etc. So once the pandemic is over, I hope some of you, if you happen to visit India, you do come and visit us at IMTech. At IMTech, I have a lab of molecular glycobiology, but before I go into the recent research which we've been doing, I thought I would take you through my scientific journey. So first of all, when I started looking back at my journey, I realized that my scientific journey has been more like a leaf blown in the wind rather than an intentional pursuit to start a career in science. So I'm really grateful to all the forces and the people who nudged me into this career. I'm very fortunate to be born into a very loving middle-class family. My father and grandfather were in the legal profession, and my father retired from the Indian judiciary, from the High Court of Madras in Andhra Pradesh. My mother gave up her job in banking sector to look after us, and she was especially very involved in our schooling. And my elder sister, younger brother, and I grew up understanding the importance of academic excellence and education. So we were really very good students. Despite that, I really didn't have an intention to do science because I didn't really know what scientist profession is about. When I was small, actually, my earliest childhood ambition was to become a dancer, probably because my older cousin was a dance teacher, and also because I was then interested in different hairstyles and dressing up. But once I was older, my elder sister, who has been a very big influence on me, she has always been a wildlife enthusiast, and she's now a professor studying elephants in an institute in Bangalore. So because of her interest in wildlife, I also read a lot about animal behavior and wildlife, and I thought I was interested in that too. However, I was still not sure what to pursue. My younger brother was very interested in tinkering with different machines and fans and clocks and things like that, along with my engineer grandfather and uncle. And I realized that I wasn't interested in that. So that sort of negative selection sort of helped me. After my 11th standard, we moved to Hyderabad because my father accepted a transfer there. And that also helped because because of the transfer of schools, I was not eligible to try for engineering and medical streams, which I was anyway not really interested in. So I followed my sister's footsteps into the St. Francis College at Hyderabad, where I did my bachelor's degree in chemistry, botany, and zoology. This was actually a course which had equal emphasis on all three subjects. And I enjoyed organic chemistry a lot. And I also enjoyed studying about different systems, organ systems in plants and animals. At the end of my BSc, though, I was still not sure what I was going to do. And I even toyed with the idea of doing a master's in journalism because I was very interested in writing. But fortunately, I got to enroll at the University of Pune for an MSc in biotechnology. And I was quite excited then because this was an upcoming field then. Now, I'm very grateful to all my teachers because they're really very good in teaching me different things and values too. But the system of education was such that there was more emphasis on memorizing rather than on scientific questioning or on learning concepts, scientific concepts. But at my MS, when doing my MSc, I had a very good mentor, Professor Sitaraman, who really opened my eyes to the world of research. I then realized what doing research was actually about. I worked with him for a dissertation on mitochondria. I also had a very wonderful teacher who taught us NMR, Professor Ganesh, from the National Chemical Laboratory of Pune. And I was so interested in the NMR that following my MSc, I decided to do a PhD working on NMR of proteins to look at interactions with ligands. So I was fortunate to get to be a PhD student at the Molecular Biophysics unit at the Indian Institute of Science, Bangalore, which is one of the premier institutes at India, with Professor Surolia, who's a very well acclaimed scientist and especially a glycobiologist. So we were planning to do NMR spectroscopy to study the interactions of winged bean agglutinin, a lectin from a winged bean, along with Professor Siddharth Sharma, who had joined Indian Institute of Science then, and who was an NMR expert. But at that time, because of not having everything together in place, especially with respect to the instrumentation, we had to abandon this project, and I instead started working on the malaria parasite Plasmodium falciparum a year after I joined my PhD. So even there, I was first planning to work on heme biosynthesis enzymes, but after a year, realized that the enzymes which we were expressing were not really active. So, but the third time we were lucky. The third project which I started on was on looking at the kinetics of death, which is caused by different kinds of anti-malarial drugs. And I showed during my PhD that there are inhibitors of a plastid organelle in Plasmodium falciparum. And if these inhibitors actually inhibit housekeeping functions, then they have different kinds of they have a different kind of death kinetics as in comparison to inhibitors which actually inhibit non-housekeeping functions. I also went on to look at the mechanism of action of an immunosuppressant, deoxyspergualin, which also happens to have very good potent activity against the malaria parasite. During my PhD, this was my first paper, a review article, which actually came about from a presentation which I gave during my coursework, which Professor Surolia actually, he encouraged me to write it up as a review article. So it was interesting. So I was quite confident of my writing skills, and I was actually very proud of the way I had written up this review article. When we sent it out to an Indian journal, Current Science, and I got back to review, I was quite shocked because I looked at the kind of criticism which was there. And it was my first experience looking at a peer review of an article. So that, of course, then Professor Surolia explained that that is how a peer review is, and you need to take review constructively and not take it personally. So that was a lesson which stayed with me. I learned a lot during my PhD, and most of my credit goes to my mentor as well as to my lab mates who taught me many of these skills which I picked up then. Of course, being in a lab of a glycobiologist, I also was involved in many other projects which were to do with lectins. And I also attended a glycobiology conference where I met Professor Jim Paulson, who offered me a postdoctoral position. So following my PhD, I went to the Scripps Research Institute and worked with Professor Jim Paulson. At Scripps, I worked on sialic acid binding lectins. I used chemical biology techniques to visualize glycoconjugates on cells and do proteomics of these cells, and I also glycoproteomics. And I also used photo cross-linking techniques which were developed at the lab to look at ligands, the binding partners of Siglec called CD22, which is there on B cells. My experience at Scripps was quite different from my PhD. First of all, the research was so fast-paced. It was so nice to get to do research where it was not limited by waiting for a reagent to arrive or waiting for funds to arrive. So it was really very nice to work at Scripps. And then another thing was working with Jim really taught me how to present data effectively, how to also write projects, how to look for impactful science. So I'm really grateful to Jim for mentoring me during my postdoctoral days. I was also fortunate to meet my husband during my postdoctoral tenure and get married to him. After my postdoctoral tenure, when we were looking at returning to India, we actually tried applying in many labs in the southern part of India because our families live there. However, we also happened to interview in Chandigarh, and we were lucky to both get positions here. I realized how fortunate we are in that sense. And so in 2010, we joined the Institute of Microbial Technology, where we work. In looking back, I realized that many of my traits I have actually, which are required for doing science, I am indebted to my family. I learned to be disciplined from my mother. I learned to be imaginative and be positive from my father. Conscientious from my sister, and to be clear in thought and positive from my brother. I am also, after my family, I'm really indebted to my mentors, my teachers, my lab mates, and my friends who have shared my research journey. I also realized that I have learned to believe in myself from my husband, and to be curious and enthusiastic from my 2 daughters. This is a poster which I remember from my PhD days, and which my students also have in our lab now. This really resonates with me, and so I thought I would show it here. And I also like this poem, and this actually helps me to keep sane when things are not going as well, when we have challenges in funding or with with research projects really taking off. Now in India, the pace of research is not as fast as I experienced in the US. But I also realized that there is also something positive about it. We have a lot of quiet time, and this quiet time we can use to plan experiments. We can use it to look at how far we've come or how far we want to go. We can also use it for writing up projects, for writing up data. So in all, I'm really grateful for this opportunity to be here and work here in INTEC. So the lab which I've set up at INTEC is a molecular glycobiology laboratory. We work on various aspects of glycobiology. We work on lectins, which are glycan-binding proteins. There are some microbial fucolectins we work on. We've also worked on human intellectins in the past. We've also worked on metagenomics analysis, looking at carbohydrate active enzymes in the human gut microbiota in Indian individuals. We've also worked on alginate lysis in clearing Pseudomonas biofilms. We've recently started a project on looking at glycosylation of SARS-CoV-2. And we also work on sialotransferases and trying to use them in synthesizing human milk oligosaccharides. And we work on fledgling non-bosonic acid transferases. For all of this work, all the credit goes to my PhD students. Without them, none of this would have been possible. I thought I would share with you a little bit about about our work on fucose-binding lectins, and then also talk to you about fledgling glycosylation. So why are we interested in glycan-binding proteins? Now, if we look at cells, there are different types of biomolecules. Now, most of the work which is going on in molecular biology is usually on proteins because of the ease with which we can study proteins because of the template-dependent synthesis from nucleic acids. But if we look at cells, we can also see that while we have a lot of proteins, we have lipids, we also have glycans, and glycans make up usually the extracellular portion of the cells. And they are actually the molecules through which cells actually contact each other, through which they signal. So when we're looking at glycans, we can see that glycans are not synthesized in a template-dependent manner, and they are actually synthesized by glycosyl transferases. And because of this non-template dependent synthesis, we see that there is a lot of complexity. So if you look at the complexity of even a single complex glycan, you can see that you can have as much as 100,000 different variants of this kind of a complex glycan. So how do we actually study such complexity in glycomics? One way to do this is by looking at glycan-binding proteins. So glycan-binding proteins actually break down this complexity into distinct functional glycan motifs. So they offer a handle by which we can study different kinds of glycans. When I joined the Institute of Microbial Technology, I wanted to have something to do with microbes. And so we decided to first look at fucose-binding lectins. This is a class of lectins called the F-type lectins. And these lectins were studied earlier in vertebrates. They were studied in invertebrates too, but there was very limited knowledge of them in bacteria. So we started with bioinformatics analysis of this group of lectins, and we realized that they were very, very widespread in bacteria. They're found in different kinds of classes. This is phylogenetic tree, which shows the F-type lectins in different classes of bacteria. And they were also present they were present in the form of chimeric proteins. So they were actually present with different kinds of enzymatic as well as non-enzymatic domains. So this really got us thinking about what they might be doing in the context of so many different kinds of domains. If we look at this F-type lectin, it is basically a smallish domain. It is a beta barrel which has loops. These loops actually form variability patterns, and these are the ones which are involved in binding to fucose. We found in the lab, we realized that we could increase the affinity of this F-type lectin by actually expanding this barrel and creating a wider interface at which they can oligomerize, and because of the higher oligomeric status, they are able to have higher avidity and therefore better binding strength. We also have studied this fucose binding lectin with regard to its effect on its tandem fucosidase domain. We saw that this tandem fucosidase domain has much better enzymatic activity as compared to a fucosidase domain which is not present along with an F-type lectin domain. And we proposed a model of a bind and jump wherein we think that this substrate, the fucose substrate, actually binds to the fucose binding F-type lectin, and then it jumps into the fucosidase domain. And this actually helps the fucosidase domain to recruitate better. And once the fucose is cleaved off the substrate, the fucose can go back into it is actually binding back into the F-type lectin domain, and that prevents product inhibition, feedback inhibition. We have also looked at the different active site, the binding site residues of the F-type lectin domain, and looked at the variability in different kinds of F-type lectin domains from different bacteria. And recently, we have also studied an alginate lyase domain, which is present along with an F-type lectin domain, and we have looked at its potential in clearing Pseudomonas biofilms in cystic fibrosis. So this study is going on now, and we are trying to see how effective it is in the lung environment in cystic fibrosis. Now I would like to show to you a little bit about another line of research, fledgling glycosylation, which we have been studying. Now we know that glycosylation is very, very prevalent in eukaryotes. It is the most abundant kind of modification which is present. Now in bacteria too, it is more and more apparent that there is a lot of glycosylation. And glycosylation in bacteria is mainly of the N-link type or the O-link type. Of the O-link type, we can see that there is one of the types of O-link glycosylation which is there in bacteria is actually glycosylation with a non-ulosonic acid. And this has been studied in gram-negative bacteria. It has been studied in Campylobacter, Helicobacter, Aeromonas, and Mangroves. And the studies which have been done have actually shown that there is a glycosyl transferase called MAF, which is required for glycosylation of the flagellin. And it is also known that if you knock out this, if you delete this MAF, glycosyl transferase gene, then you do not have bacteria which are motile anymore. They lose their flagella. And this is despite the glycan being synthesized. And this was the evidence that actually led to researchers realizing that this MAF glycosyl transferase must be required for flagellin glycosylation. It is required for transfer of the non-ulosonic acid onto the flagellin. Recently, there has been also structural study of MAF glycosyl transferase from magnetospirillum, and this has actually shown that this domain looks a lot like silent transferase, so it is probably indeed a glycosyl transferase. However, at the time when we started this work, there was no, even till now, there was not much known in the terms of this glycosyl transferase in gram-positive bacteria. what was known is that these the kinds of glycans which are transferred onto Clostridium botulinum is actually legimic acid, which is a non-masonic acid, a 9-carbon sugar, which is very similar to sialic acid, N-acetyluraminic acid. And from Clostridium, flagellin was shown to be glycosylated, with a derivative of legimic acid. And we thought that if we could actually use this MAF glycosyl transferase of Clostridium, we might be able to actually transfer sialic acid and make a new linkage of O-silylation. So with this aim, we were starting to study MAF glycosyl transferases. And then there was this paper by Bimar et al. Who showed bioinformatics analysis of the flagellin glycosylation islands in different strains of Geobacillus. And in one of these strains, we saw that this MAF glycosyl transferase was actually present along with biosynthesis cluster of N-acetyluraminic acid. So this actually indicated that probably the MAF glycosyl transferase of Geobacillus is involved in actually transferring a sialic acid rather than a legimic acid or a sodaminic acid onto the flagellin of Geobacillus. So considering this, we decided to study the MAF glycosyl transferases of Geobacillus and of Clostridium. To study this, we made use of an E. coli EV136 strain, which was made by Professor Wimmer from Illinois. This strain actually is actually a K12-K1 hybrid strain, which can make a polysialic acid capsule. And the new S enzyme, which is required for basically polymerizing sialic acid into polysialic acid, is knocked out here and therefore is deleted over here. And so what we have is an accumulation of CMP sialic acid, which is the substrate of MAF of silyl transferases. So we decided to use this system in order to overexpress our MAF glycosyl transferase as well as the flagellin and see whether we could see any glycosylation of the flagellin in this system. We also used a regular lab strain BN21D3. Now BN21D3 does not actually overexpress CMP sialic acid, but it does have CMP KDO because KDO is part of the lipid. And therefore we thought that if our MAF glycosyl transferase could transfer non-losonic acids, we could also see whether it is able to transfer octilosonic acid like KDO onto flagellin. Using these systems, we actually looked at Geobacillus MAF expression and Geobacillus flagellin expression in these E. coli strains. We were able to co-express both of these. We were able to show that using a sialic acid biotinylation strategy, that actually when we co-express both the MAF and the flagellin gets biotinylated, meaning that it had sialic acid. And we were also able to show by mass spectrometry that the flagellin, which is seen as a single peak here in the intact mass, when it is co-expressed with MAF, we can also see the peaks which are modified with the 291 Dalton, which corresponds to the mass of sialic acid. We could also do tandem mass spectrometry to show the glycosides on the flagellin, which were actually modified with sialic acid. Similarly, we could also show that Clostridium botulinum MAF also is able to transfer sialic acid onto Clostridium flagellin. We could show that flagellin is modified with sialic acid when it is co-expressed with MAF. The intact mass analysis also showed many peaks which are different by 291 Dalton, which refers to this mass of sialic acid. And tandem mass spectrometry also could show us the glycosides on which flagellin gets glycosylated with sialic acid. Now, if we look at flagellin, this is the cross-section of a flagellar filament, showing the arrangement of the flagellin subunits. Now, each flagellin has these different domains. In the gram-negative flagellin, we see there are D2 and D3 domains, which are actually the ones which have been shown to be glycosylated. In gram-positive flagellin, these domains are actually not there. So the Clostridium and Geobacillus flagellins look more like this without the D2 and D3 domains. When we looked at the mass spectrometry, tandem mass spectrometry data, we could see that the sites which were glycosylated actually correspond to the D1 loop in the gram-positive flagellin. We could also actually quantitate the amount of flagellin glycosylation happening and in E. coli when it is heterologously expressed, and we could see that as much as around 50% was actually glycosylated. Apart from this E. coli strain, which was overexpressing CMP salic acid, we also co-expressed the flagellin and MAF in E. coli BL21D3, which has CMP KDO. Also in EV36, which is another strain which doesn't overexpress CMP salic acid so much. And in both these strains, we could see that instead of salic acid, we now see KDO, which is modifying the flagellin. We could actually confirm the identity of the flagellar glycans by doing a mild acid extraction and tandem mass spectrometry and comparing with the standards. And we could see that actually it was indeed salic acid and KDO, which were modifying the glycans in these systems. So this work we have recently published in Glycobiology. I would like to end my talk with acknowledging all the people in my lab. So Ritika, Shelza, Gurdeep, Akhil, Asavri, and Sonal are past PhD students. Sonal, Asavri, and Ritika were actually, and Sonali were involved in some of the work which I have been showing you. Asavri and Sonali were involved in the flagellin glycosyl transferase work. Ritika, Sonal, and Madhu and Neetu all work on, and Asavri too also worked on the F-type lectins. And the other students in the lab who are working on different aspects of glycobiology, which I mentioned earlier. Dr. Babu from C-Camp was also involved in the mass spectrometry analysis in the work which I showed you now. We are grateful to Professor Wimmer for providing us the E. coli strains, without which we couldn't have done this work. We are also grateful to the mass spectrometry facilities at INTEC, at Taplin, and at IIT Ropar for the mass spectrometry analysis. The Consortium of Functional Glycomics for the F-type lectin glycan binding profile, and our funding agencies, CSR MTech, as well as SERB. And I am very grateful to my mentors, teachers, friends, and family, for they are because of whom I am here today. And thank you again. If any questions, I'm glad to answer.
So thank you very much, Dr. Ramya, for sharing your journey from very young age until a professor in India. So thank you very much for sharing that. So my first question is this: as a PhD graduate from India and going or finding a postdoc in other countries, and as a woman, have you encountered some challenges in that direction? And so, how did you solve that challenges and problems on your finding postdoc in the US?
Thank you, Doreen. So actually, I was very lucky that I have a very supportive family who encouraged me to do whatever I wanted to do. And with regard to finding a postdoctoral fellowship, I really didn't face any challenge because I just happened to be working in Professor Surolia's lab, and he's a very accomplished glycobiologist. And he actually organized a conference to which Professor Jim Paulson was invited, and he attended. So I didn't really have to go looking for something. I was really lucky, that's what I'm really lucky to land a postdoctoral position over there without even having to look for one. So I cannot really talk about that, but I'm sure others do face challenges. There are, India has many PhD students. It is very competitive. And I'm sure there are many of my friends too did have to actually apply for quite a bit of time before they could find a suitable postdoctoral position.
Okay. So we learned that your husband is also a scientist. So how did you balance work and home in that aspect? Both of you are busy with work and then you have kids. So as a woman scientist, how did you balance that? Please tell our young researchers who are also in the same position as you are, so that they will also get some wisdom from you.
Right. First of all, I'm very grateful because my husband is all for women empowerment, and he also helps to make sure that I can do well in the lab. So of course, having said that, here in IMTech, we don't have any organized daycare or any such child care facility. So when we, my elder daughter was born soon after we joined here. So in the beginning, it was quite a challenge figuring out how to balance, how to make sure that we would be able to work as well as look after our children. So I'm very grateful that my family helped. They came here, and my parents and my in-laws, and they helped to make sure that we had a smooth transition till we found a babysitter, and who also was a very lucky find, and who has been helping us since then. And during this COVID time, my parents are here too, and so that has also been a great help. So we have managed to find a support system. I'm sure everybody finds it in their own way. So that I guess that is the crux to really being able to manage your work along with raising your family.
Okay, so we have one question here from Tai Min. Can we deglycosylate bacterial glycoproteins? How? Can we deglycosylate bacterial glycoproteins?
Can we deglycosylate? Okay,
yeah.
So you can glycosylate proteins using PNGase, which removes the N-linked glycans. And you can also deglycosylate O-linked glycans using beta elimination using alkali. So there is published literature which would be able to, but there are of course certain linkages which may not be amenable to deglycosylation with PNGase-F or PNGase-E. So that might require some, I will have to know which particular glycan they are referring to. But in general, N-linked glycans can be deglycosylated with PNGase, and O-linked glycans by beta elimination.
Okay, so thank you very much. So I have a question for our three speakers, and this question is this: Our training to become where we are right now as professors in science is very long, particularly for a woman. From primary school, secondary school, to bachelor's, to master's, to PhD and postdocs. And sometimes there are some things as a woman that we already neglect because of this long journey. So is this always the direction, or is this journey a recipe to become where you are right now, or there are some aspect in that direction that you could forego and be a successful woman professor? So Dr. Ramya, can you answer that question?
From my experience, I know that this journey was essential for me. Without my PhD, I would not have really understood how to go about doing research. And even the postdoctoral research really shaped my independent science skills. So I think, at least from my perspective, I think for PhD as well as a good postdoctoral tenure is required to hone your researching skills. But I do know of a few scientists who have been able to start off their independent career directly after their PhD. I think it depends on the level of skills which you have obtained during your PhD, and also how much exposure you have had to different kinds of research during your PhD. I'm sure Professor Jessie and Professor Montero may be able to share much more in this regard.
Okay, thank you. Professor Montero, can you say something on that?
Oh, it's like, are you asking, is that a shortcut, not
Yes,
a long journey?
Yes.
Yeah, I think it's just like Ramya. I don't see a shortcut here, because you gain so much experience. And, sometimes, for example, the cell biology in the US, I never get to use, but at one point, I think I start to dig up that knowledge now. So no, for me, I think there's no shortcuts, but you just enjoy along the way. I always think back of the sweet time, and I think it's you growing up slowly like that. Yeah, I think you just have to enjoy the path. And that's my thinking. Maybe Gisela, Professor, you have any idea?
So, well, unlike you, I haven't shared yet some personal facts about myself. So, first of all, I lost my mother when I was 5. So I grew up without a mother. So my father raised us siblings, and he was a very strict disciplinarian and valued academics. So we had to grow up excelling in our studies since we were in grade school and high school and all that. And then my father, who was a medical doctor, wanted me to be a medical doctor. And so I kind of tried that after a bachelor's in chemistry, which I pursued because of a high school teacher, an inspiring one, a good one. Okay, so then I tried it. I actually went to UP Los Baños to teach, and on the side I was taking zoology. You may not know that, Gladys. Preparing for med school, because my family is like a family of doctors, lawyers, no scientists in my family. But then I realized I didn't want to go to medicine, so I stuck to chemistry, and I started enrolling in my master's. But then suddenly, being young and you might say vulnerable and impressionable, I met my husband, my future husband, and I got married young. Okay, and then I started our family. So no shortcut, Montero. It's really just having to be patient and learning it from your family values. So I did a lot of part-time. And so I finished my PhD quite late compared to you, because I have 5 children. Okay, I have 5 children. And as in your societies, we do have extended family, your own family, your in-laws. And then we actually have also, we have to say it, we have helpers, extended family who are like family to us, but not related. So they help take care of the children. Okay, so in my case, I value so much my experience and my insights as a woman. So there's something different about women. The way we think, the way our brains are wired up, and we're really more of multitaskers. And we extend this to our labs as we do it to our own family. So as a mother, as you mothers, say you have more than one child, you treat each child differently. Okay, you accept the talents and maybe some of the deficiencies of each child, and try to encourage each child to develop himself, herself to the fullest. And like me, I was not forced to become a doctor. I was asked by my father to pursue what I wanted, be a teacher, not earn a lot, and things like this. It's okay as long as you're happy. It's the same, I think that I did with my children. But what I'm trying to say here is this kind of patience and tolerance or great sensitivity to humans is possessed by women. Okay, ever since the start of civilization, it's the women who brought the family together or kept the family together. and made the family, the children successful. But on the other hand, we as women, our challenge is, are we for nature, fulfilling our own nature, our own talents, or are we for nurture? Are we for rearing the next generation of talented people, our children? And so we need to balance this and say, wait, I have to make my own contribution to society and the rest of nature, because I'm not even sure that my children will do it better than me. Okay, so that was always in my mind. I need to do things by myself, for myself, pursue my science, even as I was trying to share this experience with my children, not nagging them to be scientists, but also to just pursue this path of hard work, of a discipline, of trying to be sensitive to what's around you, to the problems around you. So I actually have two daughters. So one is a postdoc in MIT, okay? And the other one is a medical doctor, okay? And she's already she's a nephrologist. She's the head of the liver and transplant program of Vanderbilt University. But Carla Gisela, she's my scientist daughter. She's studying cancer and doing the whole range of, proteomics all the way to in vivo. So I guess you also have to, share or lead by example. And so this is the big thing for me. I consider my mentees, my RAs, my students as my family. They're my non-biological family. They're my academic family. And it's the same thing I would do in the lab, hold lab meetings regularly and, try to follow up on each one's project. And they think that I don't remember what they're supposed to do, but I do. And it's got to be this kind of a one-on-one thing, a treatment with your postgraduate students and your research assistants. So it's really, university is really this breeding ground for so much talent, okay, in the world. And we know it. That's how our civilizations grew anyway. Our cultures grew because of our universities. It started in academia, in Socrates, Plato, Aristotle. At that time, it's all just thinking rationally, Q&A. But then by the medieval times, then experimentation was growing. And so, you needed to get the best and brightest students into the universities and, take good care of them like your own biological family. So I think Asian women in particular know this so well. Their role as mothers, wives, and now as teachers and research mentors. It's so critical for all of us to think this way.
All right. So thank you very much for your wisdom and sharing. So I think despite of race, whether you are from India or from Thailand or from Philippines or any other countries, there is always common denominator for all. And that is, as a woman scientist, we are there to nurture and support our lab in order to come up with the next generation scientists. Yes. So we have here one comment from Professor Alina Sir Alfad Yusuf. She said that as the head of our Gender and Development Office of Zamboanga State College of Marine Science and Technology, Zamboanga City, which is south in the Philippines, all information and inspiring stories I've heard today from our very motivational and competent research persons will be shared to all emerging young scientists. Very timely event, especially that Women's Month is fast approaching. Kudos, everyone. So that's her comment. Thank you very much, Professor Yusuf, for that kind comment. So we have here one question for the three of you. During this COVID-19, from Mukesh Sikarwar, I came to know about lots of disagreements on scientific data and outcomes. I would appreciate if any scientist here would like to answer my question, which is how to trust which research is unbiased or results that are not influenced? So any one of you can answer that. So which research to be trusted, especially during this COVID pandemic? Is it the research that is unbiased or research that are not, or results that are not influential? Or yeah, results that are not influenced, whether by politics, perhaps by politics or what?
Maybe we'll get Montero and Rana to answer first. Get to yeah, say a lot of things on that myself.
Professor Montero, anything?
Wow, that's a tough question, you know, yeah, because it's hard. I think, you know, Dick Anderson used to tell me that, you know, the good result is the result that is reproducible in a different context. No, not repeat of the same with the error bar, but it reproducible. So I guess it has to be two sources, and if it confirm each other, I guess you try to trust more than one sources, you know, I guess. Maybe Ramya, you have anything to answer?
Yeah, I guess we need to look into the data carefully and see whether it is really reported completely open. And we trust data that, I mean, we expect it to be reported with integrity. So if it is if all the data are shared and open, then I think we can make a decision based on the quality of the data.
So I like to say that the situation is very different from the normal, you know, scientific environment we're used to. So this is what you call force majeure times. Okay, so these are unusual times, and so scientists, well-meaning ones all over the world, are really trying to find ways to contribute to solutions to our current problem. And you cannot fully blame some for publishing their data quite prematurely. And it's now been pointed out that preprints have flooded the scientific literature, and some of them are not, well, turn out not to be fully validated. And recently there was an article in Science which pointed out, you know, all the facts which needed to be checked, the results that needed to be checked. And in fact, very prestigious journals, medical journals, The Lancet and the New England Journal of Medicine, had published some studies that didn't turn out to be, to withstand the test of high quality medical science. But then I think I tend to be more forgiving during these times because of, you know, force majeure. I don't know that anyone is trying to unduly benefit from too much publicity or publication or even any financial gain. But on the other hand, you're right. We need to read the articles in our fields of specialization and evaluate them for their validity or for their high quality. And it is our responsibility to share it with the general public. So there's this ongoing conversations even within family. So suddenly now, non-scientists, laymen in our families, our communities, are trying to understand virology and vaccine science, but it's almost impossible for them to do it. So it's our responsibility to try our best to do it. And then again, ourselves, we're not so sure. And of course, we know in science, there's no absolute truth. Science builds on the truths that it had, you know, well, discovered in the literature. And it's for us to continue constructing the truth to the fuller truth. Okay, so in the PASE, this is the Philippine American Academy of Science and Engineering, where Gladys and Doralyn and many of our viewers here today belong. We decided to come up with PASE COVID-19 bulletin. So I started this the start of my term, which coincided with the start of COVID. This was in early 2019. And so we decided that among our members in our organization, depending on our areas of expertise, we asked our scientists and engineers to cite scientific evidence, published work, to come up with a bulletin on 3 major strategic areas. And they're all found in our website, the paasa.org website. So the 3 areas of a strategic action would be containment and mitigation, and then triage and treatment, and third is testing and fast tracking. So right now we already have 18 bulletins going on 19 posted in the PASA website on all of these topics. And you will find scientific references underneath each of those advisory bulletins of the PASA. So it's really tough during this time. So we have to be more tolerant of scientists and the publications and even the pharma companies.
Right. So thank you very much. So this will be the last questions to the 3 of you before we end this global breakfast meeting. So in these pandemic times that will last for another year or so, how are you going to inspire the young women scientists in our participants to pursue their science career? And perhaps give them an invitation to join your lab should there be some opportunities in the future. So, Dr. Ramya?
Yeah, I am actually fortunate to be in IMTech, because in IMTech we have a sort of a organized way in which we get students interviewing for PhD positions in the lab. So actually the system is basically taking care of the students coming in. But at the same time, we also have some outreach programs where we try to inspire school students. That is actually, taking a little bit of a backseat. We cannot do those kind of activities where they visit us or we visit them, but we do still have some webinars where we can reach out to school students. Yeah, so I think that's the way forward. We will be engaging with younger audience via webinars.
Okay, so thank you very much, Dr. Ramya. Professor Montero?
Yeah, so this has been a difficult times because I have 2 students plan to come and they have to subject to this 14 days quarantine, and it's quite a lot of money if you're not Thai citizen. So at least, but we have this Zoom thing. I don't think if we don't have COVID, we wouldn't have this thing. So I try to think positive, and I would like to welcome students to come to, if you want a PhD program in Thailand, I would be happy. It's so hard to find a Thai student to enroll in the graduate program with me anymore. So my lab become more and more international. And so I would like to welcome anybody who would be interested to join me. And Ramya, I have something I want to work with you, continue. You have something that I will contact you later, and also Professor, Professor Glycosylation, maybe there's some opportunity to work. Okay, that's good. Yeah, that's it.
So much. So just listening to our 3 talks, we're all linked because I talked about enzymes as well. Okay, and I also have a publication on antibodies. So I did work with an antibody guy in the USNIH, a member of our PASA, Eduardo Padlan. And I tried to set up phage display technology in my lab, but I was just into too many things already. So by all means, Montero, please take in the students of Cheryl and Gladys and train them to bring phage display technology to the lab in UP Los Baños. You know, last 2 weeks ago, our PASA distinguished lecturer was none other than George Smith. Okay, none other than George Smith. And so here I was again, Yeah, and you know, he is a pharma economic advocate. So us, yes, about, you know, the patent monopoly and all that stuff. So he's like working for all of us in Asia. Okay, but then again, I want to point out Serum Institute of India. That's so important in getting the vaccines manufactured for the rest of the world. Serum Institute of India. So I have a very strong interest in immunology. And then I also, of course, talked about cellulases and sulfur oxidases. And we have our, you know, many seaweeds in the country that both of you are interested in. And so I also just listened again to Francis Arnold, okay, about directed evolution. I mean, you know, we're the same. We like to look at our women models. And there are just so many of them. And some of them are now beginning to win the Nobel laureate. So who knows, maybe Dora Lynn one day or Gladys will be Nobel laureates, right? Your generation. So, you know, you should dream of that. During COVID, I'd say as women, we have to count on our virtues as women. I think as women, some of our strongest virtues are what? Patience. And patience leads to what? Great optimism. Great optimism. Do not, you know, be doomsdayers, you know. So as one of our scientists, Filipino scientist, who's a priest, Father Nicanor, who did his PhD in MIT and then is now in Providence University in Rhode Island, said in his first slide, it took so many hundreds of years, 200 years to get the cure for the Black Plague. And then it took 50 years for, was it the polio vaccine or smallpox vaccine? Now it's only 2 years and we have the COVID vaccine. Okay, so I think it's so important for us to hold the fort and try to get everyone to think positively. And the way to get things done is, of course, through science. See, so how do we get the women scientists to join us? I think we should try to be role models. And that's what we're going to do in PASE. We're going to come up with materials, enriched materials, and we will make sure to include women. Right now in our labs in MSI is already managed by Lilibeth Doralyn. It's no longer by me, and I'm not supposed to go to the lab because I'm a senior citizen, right? But we do have our Zoom meetings. So Zoom is like, you know, it's really the revolutionary thing. Let's call it a Zoom revolution. It's really yeah, it's making us closer as friends and as collaborators. We're getting to connect with one another on a more personal basis. So I think it's one way of getting the scientists working together with great sincerity and trust for one another. Okay, so I think yeah, we have to make do. And our scientists, including the women, the young women, they're the ones going to our labs now in UP, Marine Science Institute, and they do the round robin. They do it on a staggered basis. But yes, they're performing experiments already. They are. So.
Right. So thank you. There you go. We have the words of wisdom from our three distinguished speakers, the women leaders in their respective field. Thank you, Dr. Ramya and Dr. Montero and Dr. Giselle, for inspiring all of us to be like you and to be leaders of science in the future. Now I will give the floor to Gladys.
Oh, thank you very much, Doralyn, for for helping me out. I apologize again to our guest speakers and to all the participants. We had power interruption here on campus. I had to go to another place. This Gani's place, by the way, Giselle, we went to his house so that I could participate in this event. Thank you. All of your talks were inspiring. And sorry, I missed a lot the discussion. I really miss a lot, but truly this will be a start of a collaboration between Ramya, Montero, and of course, Ma'am Giselle. We're always here. And one problem that we have here in the Philippines is, especially here in our campus, is we don't have enough equipment. So I think that's where Ramya can help us. She said that in their institution, it's well equipped. So here we're just starting. We're actively looking for grants so that we can start having our own equipment here on campus. So what we do is so that we can have results in our research, we collaborate with private companies like Pascual Pharma Corporation, and we also collaborate with foreign professors. So we have an active collaboration with Dr. Carlito Librilla in UC Davis. So we send our students there so that they can get trained. And you know what? When they come back, they're much better than us. We actually learn from them. So actually it was Carlito who told us this secret. You have to surround yourselves with smart people. Otherwise, you won't be successful. And we have to accept that we don't know everything, that we can learn from our students. And I do always learn from my younger students. I have mentored a lot of students who are much smarter than me, and I know they'll be more successful, but at least they have the potential. It is a good privilege and good opportunity to mentor all these young scientists who has the potential to be more accomplished than me and all of us. So thank you very much, Montarup and Ramya and Doraline and Cheryl for and Ma'am Giselle for always leading us here in Paase. She's really an inspiration to us. Look at those accomplishments. A lot of papers, but staying here in the Philippines. And you know why? It's because of collaboration. That's the thing. We have to admit that we need help, that we cannot do big projects. We cannot work on projects if we just depend on ourselves, right? So, and also what I learned is we have to reinvent ourselves. Every 10 years, we have to reinvent ourselves, I think. So I started as a synthetic chemist. I was so excited to do synthesis here in the Philippines. My first 2 years, I was so blessed because I had this very talented undergrad students who is almost done with his PhD. So they were able to synthesize compounds, but after that, it's taking me some time. So I really decided, hey, I have to do something else so that I can do science in the Philippines. So that's why I collaborated. My husband and I collaborated with Carlito. And so we're now doing glycomics and glycoproteomics. And it's good that Ramya is doing that. And we can collaborate more. And my project is not more on synthesis. And of course, I still continue that. It will always be there, although it's a little bit slow. So I'm now moving on to nutraceuticals. Okay. So we just had this talk that in Australia, they have this botanicals track for natural products. And then you can actually, if you have identified an extract, it doesn't have to be pure. Okay, it can be a mixture, it can be crude. You can apply it as a medical food. So that's one track before you go through this. So you can skip, I think, phase 1 in the clinical trial. You can go to phase 2 already. So yeah, so we really need to, I think that's how we survive in science, right? You have to be creative and you have to be resilient. And you just need to be more inventive and adapt to what we have right now here in the Philippines. And thank you again for joining us, and I look forward to have with me and Doralyn and Cheryl. I hope we will have another IUPAC Global Women's Breakfast next year, and we hope you can help. You can help us. You can introduce us to some of your colleagues, or you know what they do. We can actually have a joint GW event.
Yes.
Okay. So we can do that if the time differs. If we agree on a time, a common time, we can have that with Ramya's group, with Montero's group, and we can have our own GWB event. Yes, that's right, Doralyn. And it was Doralyn who introduced me to this event.
Right. Yes.
Thank you very much.
Yeah. So before we end, Gladys, perhaps we need to invite our participants to turn on their camera so that we can send the picture to IUPAC that we have
of course.
our breakfast.
Yes, can we request first the panelists, and then after that we'll request the all the. Do you have your camera ready? I can screenshot. Okay. All right. Can you hold on? How do I show this?
Okay.
Gallery. There you go. Okay. Oh, so only panelists can do it in webinar, she said. So the participant.
Can I suggest something? For
Sure.
ICT, can you promote everyone as a co-host so that they will be able to turn on their video? Can you do
yes,
that?
thank you. Yeah, ITC, can you do that, please? Can you make everyone as co-host? Thank you for that suggestion, Doralyn. You're an expert really in Zoom. So we'll do smile for the panelists first. Wait. I'm having a hard time. My daughter usually do this for me. Okay, smile. Okay, ITC. Oh, so there is no mass promotion? Oh, I didn't know that. So attendees cannot join? Doralyn, you're muted.
I said, can you just do it one by one? We are very patient as a woman, right? So
yes.
we will wait for the 107 participants to join us in this.
Yes, because we have to show GWB that we had more than 100 participants. This is the first, no, this is not the first time that Philippines has hosted this event, right?
Yeah.
But this is the first time in UPLB, and we encourage all UPLB staff that we should start doing this every year to empower women in science. Okay, thank you. Yes, she's doing it now, one by one. And this is not just for women in science. Those men, please don't leave. I can see them leaving. No, you have to stay. Okay?
Yes.
Okay. Okay, so yes, because
yeah,
it's
there they are.
Okay.
Wow.
There you go. These are our students, I can tell. Oh, Ma'am Aleli, it's good to
hi,
see
Aleli.
you.
So nice to see you.
Yes. So ITC, are you the one who's gonna take the pictures, or am I the one gonna take the picture? Oh, so we can have at most 100. Okay, that's fine. Okay, so we have visitors from Thailand. And do we also have visitors from India? Hi, Ma'am Aleli. Good to see you. Okay, pa-assist daw sa pag-picture, okay? I will keep on taking pictures. Just keep on smiling, people. Show your faces. Okay, thank you. Keep on smiling. Okay, show your faces, please.
So earlier, Maribel Nonato of the United University of Santo Tomas.
Yes, she was here.
Mission of NAST was here. So I think Maribel has left.
Yeah, and also Tina Binag, she was here
Tina
this
was
morning. Yeah.
I'm not sure if Evangeline Amor was here. It's a different name. She used a slightly different name.
I invited Irene. Ma'am Irene Villasenor.
Yes.
And then everybody else in our research cluster.
Yeah.
So I'm just doing.
Marge, may I introduce our chairman of the board of the Philippine American?
Yes, there's Marge.
Dr. Marge Peña is with us. Okay, but maybe well, she has to attend to other matters. But thank you so much, Marge, for joining us.
Yes. Okay, now I'm doing this the right way. I was not. Thank you for our colleagues in the Institute of Chemistry. A lot of them attended. Thank you very much. So these are these are way of celebrating Chemistry Month. In the Philippines, we celebrate Chemistry Month in February. How about in India and in Thailand? When do you celebrate Chemistry Month?
Well, I did not know of Chemistry Month celebration earlier, but
neither.
In Thailand?
I have no idea. Maybe I will check it out. Yeah.
Actually, I thought it was February for all the countries, but no, it's different for different countries. So I think it just became a what do you call that, tradition here in the Philippines to do it in.
So Vanji is with us, Dr. Vanji Amor.
Is she there? Good.
Yeah, I saw her. I saw her name in there. Yeah.
Nice. Okay, I cannot
Okay,
let me know.
Almost.
It's done?
5 more. 5 more to go.
Oh, 5 more to go. Okay.
Okay.
Let me know.
1 more to go. Okay.
Oh, there's Ma'am Evangeline. Oh, wait.
Yes.
I missed it.
All right. So everyone has the video now. Okay. Who will be taking the picture? Ladies.
I already started taking pictures.
Okay.
I just keep on moving the screen.
Dr. Migo. Dr. Veronica Migo is also with us. Thank you for joining.
Thank you.
Hello, Ma'am
Yes.
Giselle.
Hello.
Thank you, Ma'am.
Vanji turned on her video already. Dr. Amor, thanks for joining us.
Thank you, Ma'am, for joining us.
Thank you, Mamigo.
Hi, Doraline.
Hello, po. Thank you for joining.
So this afternoon, I was invited in one of the IUPAC GWB event in India. So I will be joining their event.
Fantastic.
Together with a very famous
Irene Papa also here.
Yes.
Hello.
Scientists from Australia and from India. I'll be joining them this afternoon.
Wow. So
Marian.
Yeah, Marian, Dr. Marian De Leon
Thank
is
you.
also already here.
Morning po.
Hi.
Okay. And then some of my students I can see. Okay. So did I finish? Did I take? Oh, and also another of my students, Cheryl. Thank you, Cheryl, for attending. Okay. So hold on. She said if she can stop, there no more attendees. So thank you very much. Why no more attendees? Thank you very much for attending this event, and see you next year. And please continue to be inspired and do science and be better than us. Emulate these leaders that we invited. You can do anything as long as you have the passion to do science.
Yes. Bye, everyone.
Bye. Thank you very much.
Be safe.
Thank you, Ma'am Giselle.
Good luck with your work. Bye.
Thank you, Ramya. We will
Very
contact you.
inspiring.
I will call. Okay. Thank you very much.
For inviting me. It was really nice to hear Professor Gisela and Professor Montero, really inspired by their science. And
Thank you, Ramya.
Thank you so much.
Thank you, Ramya, very much.
I had a good
Bye
time.
bye.
And I will keep contact, okay? Bye-bye.
Yes, we will contact you. Bye. Thank you.
Bye.
Leave now.
Bye. Thank you, Ma'am. Thank you, Doraline, huh? Thank you, She. She, where are you? Nasa Philippines ka? Ay, nasa